All right, so a very good morning to everyone and to our distinguished guests, friends and colleagues. Welcome to the Centennial Celebration Symposium today of the Department of Microbiology and Immunology. So together with the organizing committee for this event, we are very honored for you guys to join us today to celebrate, commemorate, right, 100 years of scientific discovery and educational excellence, right? So for today, I'm very sure all of you would have a very exciting day attending all the very insightful presentation. And so without further ado, I'd like to start today's program by first inviting Prof Chong Yap Sing, our Dean of Yong Lu Lin School of Medicine, National University of Singapore, to give the opening address. Prof Chong, please.
Okay, thank you for the kind introduction. So, guest of honor, Professor Kenneth Mak, Director General of Health, Ministry of Health. Professor Yeo Kay Guan, Chief Executive, National University Health System. Distinguished guests and ultra distinguished professors. There's so many of you, I won't try to name all of you today. Esteemed colleagues, alumni, students, and friends, a very warm welcome to the NUS Department of Microbiology and Immunology's Centennial Celebrations and Symposium. For a century, we have instilled in our students a deep and rigorous understanding of the core concepts of microbiology and immunology. Why? Well, because these are not merely academic pursuits. They are the very tools that enable us to mitigate the seismic impact of the recent COVID-19 pandemic, as well as previous outbreaks. And today, they are more vital than ever, empowering us to prepare for, respond to, and ultimately overcome the health threats of tomorrow. By examining the minuscule, we can glimpse the infinite. This is the very spirit of curiosity and discovery that has defined the department since its earliest days in 1925, when the departments of bacteriology and parasitology were founded. The department's story is one of relentless pursuit, of excellence, of discovery, and of service. Pioneers such as Professor Nripendra Kumar Sen exemplified how local research can use global impact. His groundbreaking work on brucellosis led to the discovery of Salmonella Singapore, a finding of international significance. Professor Lim Kok An achieved international recognition for isolating the flu virus during the Asian influenza epidemic and for introducing the oral Sabin polio vaccine, which played a pivotal role in eliminating polio among Singapore's children. Emeritus Professor Chan Soha transformed our understanding of disease susceptibility in Asian populations through his discovery of the Singapore 2 tissue antigen, firmly establishing Singapore as a regional leader in immunology. These trailblazers remind us that bold inquiry and dedication can change the course of medicine, both at home and around the world. As we look back on a century of achievements, let us look forward with imagination and anticipation. Today's symposium marks a significant milestone, bringing together distinguished alumni, eminent scientists, and thought leaders from around the world to share their insights and celebrate the century of progress in microbiology and immunology. I look forward to hearing about the cutting-edge research, latest advances in infectious diseases and immunotherapies, and the innovative work from our students and young investigators that will go on to shape the future of biomedical sciences. I look to the department's current leaders, Head of Department Professor Kevin Tan, Professors Paul McCarry, Justin Chu, Zhang Yong Liang, and Veronique Angeli to break new ground in infectious disease research, translational medicine, and education. May you continue to sail uncharted seas in pursuit of scientific truth. Your work, your passion remain as vital as ever today. Happy 100th anniversary to the Department of Microbiology and Immunology. Here's to a future as bright and inspiring as your past. Thank you.
Thank you, Prof Chong, for giving us the opening address. Now let us welcome our Head of Department, Associate Professor Kevin Tan, to give the opening remarks.
Thank you, Chin Wen. Professor Kenneth Mak, Director General of Health at the Ministry of Health, Singapore. Professor Yeo Kay Guan, Chief Executive of the National University Health System. Professor Chong Yap Sing, Dean of the Yong Lu Lin School of Medicine. Distinguished colleagues, alumni, and friends, good morning and welcome to our Centennial Symposium. You may have heard of this well-known African proverb. If you want to go fast, go alone. If you want to go far, go together. But do you know there's also a Chinese proverb? 众人水彩，火焰高. You know that's very difficult knowing where it came from to say that. But for those who don't know, it means when everybody adds fuel, the flames rise high. So the ideologies are similar. Great things can be achieved by collective effort. On a convergence of philosophies from very different cultures that transcend time and space. Today, as we celebrate 100 years of our department's evolution, these proverbs could not be more appropriate. Our journey began in 1925 with the founding of the Department of Bacteriology, joined in 1950 by the Department of Parasitology. In 1977, these two pillars united to create what is now the Department of Microbiology, and eventually Microbiology and Immunology. We owe much to visionary leaders such as Professor Vikar Zaman, our first head of department and an eminent parasitologist, followed by Professors Chan Soha, Vincent Chow, Michael Kemin, Mary Ng, and Nicholas Gascoigne. I'm deeply honored to serve as the 7th HOD, and it is a pleasure to see 4 of my predecessors here with us today. From the outset, our department was committed both to education and groundbreaking research, making significant contributions relevant to Singapore and the region. We tackled pressing issues such as the interactions between Epstein-Barr virus and the immune system in nasopharyngeal cancer, that's Professor Chan Soha's work, advanced the use of horseshoe crab amoebocyte lysates for detecting endotoxin, Professor Hobo and Professor Ding's work, and pushed the boundaries of our understanding of enteroviruses and dengue, this collective effort of our eminent virologists among us. Our legacy is shaped by outstanding individuals, Professor Lim Kok An, as mentioned by our dean, who led the Department of Bacteriology from 1959 to 77, was the first to isolate and describe the pandemic strain of Asian influenza virus in 1957. The work was later published in The Lancet. Professor Robert Desowitz, head of parasitology from 1960 to 65, became renowned for both his research and his candid analysis of global health systems in the malaria capers. Professor Vikar Zaman, meanwhile, was celebrated for his engaging lectures and beautifully illustrated textbooks, inspiring generations of students. In recent decades, our department has continued to attract both local and international talent at all career stages. Professor Nicholas Gascoigne, who joined us from the Scripps Institute in 2013, led with warmth and vision. Under his guidance, we adopted our current name to reflect our dual strengths of microbiology and immunology. And he was instrumental in recruiting Associate Professor John Chen, whose pioneering work on bacterial virulence in phage interactions has appeared in Science and Cell. The mentorship and collaboration exemplified by Nick and John embodies our department's core value. We go further together. This spirit of collegiality and community runs deep. Our bonds of trust make collaboration effortless. Our shared passion for science, and yes, sometimes for fermented beverages, makes this department a second home for many of us. Here, colleagues become friends, and friends become family. Our achievements are the result of not just scientific expertise and hard work, but also a culture where laboratory technologists, administrators, and academic staff unite to advance innovative research, inspire our students, and serve society. As we mark this centennial milestone, I thank all of you for being with us. Alumni, colleagues, guests, and especially our invited speakers. My deepest gratitude also goes to the organizing committee, with special mention to Professors Justin Chu and Zhang Yongliang for curating a remarkable program for us today. I wish you all an engaging and meaningful symposium. As you rekindle old friendships and forge new ones, let us reflect on the lessons of the African and Chinese proverbs. For 100 years, we have kept the flame burning, each spark ignited by teamwork, each blaze fueled by collective energy. Together, we've come far, and I'm confident that we'll shine even brighter in the century ahead. Thank you.
Thank you, Prof. Tan, for the opening remark. So now, join me to welcome our special guest of honor, Professor Kenneth Mak, Director General of Health from the Ministry of Health, Singapore.
I was warned that there will be 3 speeches here from Yap Singh and Kevin, but it's only one, so you must have taken your speeches away. It's a pleasure to be here with you, distinguished guests, faculty members, alumni, students, and friends of the Department of Microbiology and Immunology here at the NUS Yong Loo Lin School of Medicine. And it's particularly an honor to join you for this, the opening of the Centennial Celebration Symposium, as we celebrate a remarkable milestone, and that's the 100th anniversary of the Department of Microbiology and Immunology here in the medical school. And it's amidst many celebrations that the university is going through. More recently, we marked also the 120th anniversary for the school itself. So the Department of Microbiology and Immunology has stood pace with the university through that last century. The theme of today's symposium, A Century of Discovery, Advancing Microbiology and Immunology, could not be more apt. It reminds us both of the rich legacy that the department has built over the past century and the ongoing journey of innovation that continues to shape the future of healthcare, research, and medical education in Singapore and beyond. This symposium is not only a celebration, but also a platform for knowledge exchange. It fosters interdisciplinary collaboration in areas critical to human health, from microbiome, pathogenesis, and host immunology to emerging infectious diseases and vaccine development. It is my hope that the rich discussions today will inspire advancements in microbiology and immunology. Today's celebration is not just for longevity, but of transformation. A pivotal moment in the department's history came in 2015, when it was renamed to reflect an expanded focus on the critical interface between microbiology and immunology, signifying a strategic vision for the future of biomedical science. And for decades, this department has educated generations of doctors, scientists, and public health professionals, shaping how these disciplines are taught, understood, and applied in both academic and clinical settings, and has been a beacon of scientific excellence and service to healthcare. The Department of Microbiology and Immunology's contributions to healthcare and public health are equally noteworthy. Its work in developing both national and international capabilities in infectious disease response and immunological research has played an important role in establishing Singapore as a regional hub in all these fields. Furthermore, its leadership has strengthened the school's laboratory standards and reinforced the department's commitment to public health infrastructure. New research platforms for translational immunology developed by the department have bridged clinical insight with fundamental discovery, accelerating progress in diagnostics, therapeutics, and vaccines. In academia, the department has consistently led with innovation and demonstrated a strong commitment to providing continuing education to researchers, academics, and healthcare professionals. During the COVID-19 pandemic, the department launched an online educational series, Biomedical Insights into an Evolving Epidemic. And I know this because Kevin invited me to participate in one of his webinars. The webinar series shared the latest biomedical research on the evolving epidemic, including advances in vaccine science, therapeutics, and clinical management. And it was structured around the pillars of infection control, which were prevention, diagnosis, and treatment. This initiative exemplified how academic departments can respond rapidly and responsibly to real-world public health needs. The department's legacy is perhaps most vividly reflected in its people, a proud testament to its role in shaping biomedical leadership. And notable faculty include Professor Nicholas Gascoigne, the adjunct Professor Raymond Lin, Associate Professor Sylvie Alonso, Associate Professor Veronique Angeli, Associate Professor Justin Chu, Associate Professor Paul McCary, Associate Professor Kelvin Tan, and Associate Professor Zhang Yongliang, all renowned figures in public health, academia, and biomedical industry. These are just some of the leading local and international scientists who have contributed groundbreaking work published in the world's respected journals. underscoring the department's stature as a magnet for talent and hub for top-tier research. Importantly, the department remains deeply committed to nurturing local talent, ensuring that the next generation of biomedical leaders will continue to advance research and healthcare in Singapore. And as we look forward, we see a department that continues to evolve. The recent launch of the Masters of Science in Applied Biomedicine marks a major step forward in postgraduate education. This program is the first program in Asia to equip students with skills to combat emerging infectious diseases, to develop vaccines and design drugs. With a strong response and enrollment of over 100 candidates, this new program will contribute to a new impetus for Singapore's biomedical workforce and innovation ecosystem. And so, to the Department of Microbiology and Immunology, congratulations once again on your centennial milestone. And yours is a story of unwavering scientific curiosity, educational leadership, and service to society. And may your next 100 years be even more impactful than that first one. And I wish everyone a fruitful and inspiring symposium. Thank you very much. Okay, sure.
All right, so now we are going to start our symposium proper, right? So just a gentle reminder to all speakers, because we are running on a very tight schedule with a lot of amazing talks. So please do keep to your time. We have a timekeeper over there. So he will be putting up a sign saying that you have 5 minutes left, 2 minutes left, and then he'll ring a bell when you have to stop your talk, right? So we try to keep it to the time today. So without further ado, let me invite the first session chair, Associate Professor Sylvie Alonso, to chair the first session.
So good morning, everybody. It's my great pleasure and honor to introduce the first keynote speaker of this morning session and of this whole symposium. So Professor Malik Peiris from the School of Public Health at the University of Hong Kong. Prof Peiris is an eminent virologist. He has been hunting and studying emerging viruses, which are at the interface between animal and humans. In 2003, Prof Peiris was absolutely instrumental in contributing in the identification, diagnosis, and control of SARS-CoV-1. So Prof Peiris is an elected fellow of the Royal Society of London. He's also a foreign member of the US National Academy of Sciences. But Malik is also no stranger to the department. He has been interacting with many of us over the years, and we are very happy to have him today. So Prof Peiris is always telling us fascinating stories, and today he's going to share with us the avian influenza saga, past, present, and future. So please join me in welcoming Prof Peiris on the stage. Okay, so we're going to move to our next speaker, who is going to be the first speaker of the Alumni Talk series, Dr. Tan Hein Meng. He's I give you a bit of a why is he an alumni. So he completed his bachelor degree at NUS, and he did his final year project in the Department of Microbiology. After which he went to the UK to pursue his postgrad studies and completed his PhD in microbiology at King's College. And then he came back to Singapore and started his academic career at NUS in the Department of Microbiology. But the universe had other plans for Dr. Tan, and a few years after he got promoted associate professor and tenure, he decided to leave academia and to join the corporate world. So Dr. Tan today is going to share with us his journey as a microbiologist from bench to boardroom. So Dr. Tan, welcome back home, and the floor is yours. Thank you.
Thank you. Very good morning, Professor Mak, Director General of Health, Ministry of Health, Professor Yo, Chief Executive, National University Health System, Professor Chong, Dean, NUS, Yong Lulin School of Medicine, Professor Tan, Head of Department of Microbiology and Immunology, distinguished faculty, alumni, students, and friends of the department. It is a tremendous honor for me to be here today at the momentous 100-year milestone for the department that shaped so much of who I am. When I first stepped into the labs of this very department as a young undergraduate, you may not believe it, but more than 40 years ago now, I had no idea how this place and the people in it would guide my path, from pipetting cultures to leading commercial strategy in a boardroom. I can still remember that fateful day when I entered the office of Professor Vika Zaman, then head of the microbiology department at the Sea Point Lines campus, eager to find out what I could learn from microbiology as part of my undergraduate curriculum. The decision to major in microbiology was to have quite a seminal impact on my life. Indeed, it was at this department that I met a fellow undergraduate in the class who eventually became my wife. Today, I'd like to share with you my journey, how microbiology trained me to think, to lead, and to navigate life beyond the lab, from bench to boardroom, and how the discipline continues to shape my worldview in unexpected ways. Like many of you, my first experience with microbiology was peering into a microscope at tiny organisms that seemed to exist in their own universe. It was awe-inspiring. But more than fascination, microbiology taught me discipline, sterility, precision, patience. You cannot really cheat a culture plate. I still recall the hours spent in the lab, troubleshooting experiments, repeating protocols, and learning from failure. My first gram staining laboratory session in particular was one where I could not seem to get the staining technique correct, with my E. coli appearing as blue rods and my Bacillus subtilis as red rods under the microscope. I had to repeat the protocol numerous times before getting it right. Upon graduating from the university with a master's under Professor Lee and Kun, I went on to complete my PhD at King's College, London, on the Singapore Public Service Commission, or PSC, postgraduate scholarship. Until then, the PSC mainly awarded scholarships for undergraduate studies. But that year, the government decided to develop the Biotechnology Center here, and PSC launched the first ever biotechnology-related postgraduate scholarship, to which I happily applied and was subsequently awarded to be trained in molecular biology, a very nascent field in Singapore at that time. At King's, I continued to apply the same rigor to my research. That resilience has been one of the most transferable lessons of all. In the biosphere, there exists a wide variety of natural and man-made aromatic compounds. These compounds contain the aromatic benzene ring, which can be difficult to break down physically. However, there are soy bacteria, notably of the Pseudomonas genus, that are able to utilize a number of catabolic pathways to mineralize these recalcitrant hydrocarbons. A critical and fundamental feature of aerobic degradation of these aromatic hydrocarbons is the formation of catechol as central intermediates from these diverse aromatic substrates. The initial dehydroxylation reaction is carried out by a multi-component bioxygenation enzyme that incorporates both atoms of molecular oxygen into the aromatic ring to form cis-dihydrodiol. This cis-dihydrodiol then undergoes a dehydrogenation reaction to form catechols. During my time at King's, I was able to isolate a fairly large 112 kilobase pair plasmid and show that it contains the genes for the degradation of this compound benzene. I named this plasmid PHMT112. It was the first time that such aromatic genes were reported to be on a mobile genetic element. While in the laboratory, I was using molecular biology to unravel the ability of bacteria to degrade hydrocarbons in the environment. But back at the family, I found that it was still the age-old recombinant technology that was yielding results of a different kind. So at the end of my studies, I found myself returning home with 2 degrees, a PhD in microbiology and an infant son in my arms. Upon my return to NUS, now as an assistant professor, I continued my PhD work and was able to further sequence and characterize the second enzyme, in this case cis-benzene glycol dehydrogenase, which turns out to have rather unique biochemical properties. We found out that the initial set of catabolic genes was carried on a transposable element. And for the second time in my scientific career, I was involved in naming a genetic element, and this time it was called transposon or TN5542. We now know, of course, that transposable elements make up a significant portion of the human genome and are a major source of genetic variation. They are particularly important in the brain, affecting neuronal plasticity and development. And on the other hand, insertions of such transposable elements can also lead to genome instability, further contributing to disease development. Our work was regularly referenced by collaborators in the field and helped to create the current knowledge of such gene organization and their transfers among bacteria through transposons, which explain the ability of these organisms to exist in such a trying environment. And the genes that we were responsible for elucidating were called the BET gene cluster. But my group and I took a step further by incorporating this bacteria into a consortium of microorganisms that were used to treat an experimental plot of contaminated beach land at the Raffles Lighthouse following the massive Ivory-Coast oil spill in 1997 here in the Straits of Singapore. So a few of us from different departments, the Department of Chemical Engineering and Microbiology, were working with the Maritime Port Authority of Singapore to help mitigate that contamination. Around this period, I was sent as part of a study group by the university to overseas research institutes and universities to learn and design the Institute of Molecular Agrobiology, or IMA for short, which was subsequently founded in 1995. IMA has since merged with the Institute of Molecular Cell Biology in 2002. The experience I acquired designing IMA came in handy when I had to rebuild my current laboratory at Kemin, Singapore, and also the initial research laboratory in our China operations in Zhuhai. While I was settling quite nicely into the life of a tenured associate professor of microbiology here at the department, 7 years after returning here as a faculty member, my direction in life was to take a rather abrupt change. I received a call from what is possibly then the largest executive search firm here in Singapore to take on the role of the research and development director of a US animal feed company. So the talk by Professor Peiris really is very close to my heart. I must admit that at that time, I had absolutely no interest whatsoever. Firstly, because I was already tenured, and secondly, I haven't had a clue what feed additives do. But visiting the prospective lab and the firm revealed that there was something I could actually do to help the company. Also, at this time, the Prime Minister then, Mr. Goh Chok Tong, challenged those working in a university with a PhD to step into the corporate world and contribute towards the economy of our nation. My heart was stirred. One thing led to another, and before long, I left the corridors of this great institution to join Kemin Industries. I thought I'll show a little bit about my company. This company was founded in 1961 by both R.W. and Mary Nelson. The founder, R.W. Nelson, unfortunately passed on 2 months ago at the age of 98. This is a family-owned business, employed about 4,000 employees worldwide, and we have business operations in more than 90 countries. Our annual revenue exceeds $1.5 billion, and this is really a company that is founded on science, and so the emphasis on patents and research innovations is very strong. Just an idea of where we operate around the world, and in my current role, I'm responsible for the business in Asia-Pacific region, excluding India and China. We are the Asia-Pacific headquarters, manufacturing our products here in the north at Senoko campus and exporting to all these markets. In animal nutrition and health, our vision and our belief is that we need to raise healthy livestock and poultry. And Kemin delivers products and services to achieve this end. As a company, we hold yearly scientific meetings where our latest research and innovations are presented to the company, as well as a panel of scientific advisors who are all of them experts in their respective fields. Up until the year 2000, antibiotics have been regularly used at subtherapeutic levels to help animals stave off infections. The use of such antibiotics also led to better growth and productivity of food animals such as poultry and pigs. However, it was also becoming increasingly evident that the use of such antibiotics was leading to widespread antibiotic resistance among the human population. The European Union has since 2003 banned the use of such AGPs, or antibiotic growth promoters as they are called. My first task at Kemin was the challenge of finding an alternative to replace the use of AGPs. One of the pathogens that becomes important when antibiotics are removed from the diet of broilers and pigs is the pathogen Clostridium perfringens that can cause necrotic enteritic conditions, leading to high mortality of the flocks. The research group and I started by isolating bacteria from healthy chickens that could have probiotic properties. We discovered such a strain, which we identified to be Bacillus subtilis PB6. And so here is a slide showing the anti-Clostridia activity against all the important Clostridia pathogens, perfringens, septicum, and difficile. When we tested this against the Clostridium isolates obtained from the field, PB6 showed effective in vitro anti-Clostridia activity. we were able to isolate and characterize the active ingredient and found it to be a surfactant, a 7-membered cyclopeptide comprising both L and D amino acids. Surfactant is an antibacterial lipopeptide that is a powerful surfactant. It has multiple biological activities, including antiviral, antimycoplasma, antiprotozoa, antibacterial, and antifungal. But we view surfactant as one of the possible alternatives to AGPs. Its mechanism, among others, lies in attacking pathogenic bacteria's cell membrane. And here you see this micrograph showing its ability to cause holes in the cell membrane of the organism, causing lysis. Incidentally, these pictures were generated by the electron microscopy lab here, led by that time by Professor Malik. So this has found its way also in peer-reviewed journals. To test the effectiveness of this probiotic PB6, the product that we have created is called Closted. In controlling necrotic enteritis in broilers or chickens, we conducted a disease challenge model at Georgia in the United States. What we did was to create a subclinical necrotic enteritis condition. And you can see from here that under no disease challenge, this is the weight gain of a 27-day-old broiler. Typically, you raise chickens to about 35 days, and they are about 1.8 kilograms. And the ones that you eat in KFC are normally much smaller. The feed intake is an important parameter. And finally, is what we call the feed conversion ratio, because this has not only productivity considerations, but it's also important to the broiler raisers because it is about profit and loss. And of course, you can see that under this negative challenge control, the mortality was very high. It's about more than a third of the flock was wiped out by this condition. To this control was added the antibiotics that is routinely used, which is called zinc bacitracin. And you can see straight away there's an improvement of the, this positive control had no disease challenge. I beg your pardon. So this is disease challenge. This is no disease challenge. You can see that the background is very clean. To this disease challenge was added zinc bacitracin. And you can see that we have the mortality to about 17%. There is improvement in the weight gain, back to about the positive control, which has no disease challenge. Feed intake was more or less the same, and the feed conversion ratio was more or less the same. So zinc bacitracin is doing its job, and it is really the antibiotic of choice throughout the industry. When we introduced our Closted to this disease group, we found that we were able to restore the weight gain, the feed intake, as well as bring the feed conversion ratio back to as per control. Importantly, our mortality rate was even half of the antibiotic treatment group. So clearly showing that this Closted has immense applications in our industry. Subsequently, we tested this product across European Union, Middle East, and North Africa in 7 million over birds. And you can see the collection of the data. The meta-analysis of this data indicate that indeed there is an improvement in the overall mortality in birds receiving Closted, improvement in weight gain, as well as feed conversion ratio. And in industry, we rely heavily on what we call ROI, or return on investment. And one way of interpreting this is that for every dollar they spent on using our product, they receive $3 back of profit. So in a sense, this funds itself. Now, we have further conducted research on our probiotic PB6, and more recent work revealed that PB6 also produced a second cyclic peptide, and this is called fungicin. Fungicin is a cyclic peptide comprising 10 amino acids. And like surfactant, which I mentioned before, has also got antifungal properties. But what was really unexpected to us was that we found that fungicide also has quorum quenching properties, especially for Clostridium perfringens. You see, Clostridium perfringens is a commensal with low cell density in healthy animals. Perturbation of the microbiota may lead to translocation and proliferation of Clostridium perfringens in the small intestine. When they reach a certain threshold density, beyond what we call the carriage density, the quorum sensing system or the AGR-like system is activated through the binding of autoinducers or autoinducing peptides called AIP that then bind to this AGRC-like receptor, which triggers the downstream gene expression and leading to expression of virulence genes and toxic genes for host colonization and invasion, resulting in the condition of necrotic enteritis. What we found was that fungicide, which was a quorum quenching molecule, actually was able to inhibit the binding of AIP or the auto-inducing peptide to the arch-like receptor in Clostridium perfringens. And this is the result showing the effects of adding increasing levels of fungicide to Clostridium perfringens and looking at the expression of the various quorum sensing genes, the virulence factors, as well as the sporulation factor of the pathogen. All of these showing that at high concentrations of fungicide, they were able to completely eliminate this quorum sensing and thereby preventing colonization and expression of the NETB toxin gene of this Clostridium perfringens. In order to test the applications of the science and the product, we did multiple trials, this time in swine. And of particular practical relevance to us was when this was applied to numerous swine farms in Australia. In these farms, the current practice at that time led to common Clostridia syndrome in suckling and weaning pigs, and resulting in diarrhea observed in the piglets on days 1 to 7. And from day 8 onwards until the 10 weeks of age, necrotic enteritis was prevalent in these farms and leading to mortality of the flocks. When Closted was added into these farms, and here is a summary of the different results from the different farms. It was shown that the diarrhea dropped from 90% to 5% in one farm, and others had minimally 90 or 80% reduction. Interestingly, the post-weaning mortality also improved from 18 to 14% in one farm and halved in one of the other farms. Now, these are all very important parameters for swine raisers. And in the Australian example, this has resulted in massive adoption of this product in the industry there. As I look back, I realized that it was the Department of Microbiology then where I first began to appreciate the power of microbiology, not just in a petri dish, but in the real world. How microbes could become medicines, feed additives, fuel resources, or food preservatives. I now go from studying trial data to reading spreadsheet of a different kind. So I still routinely get to see data such as this, but I now have to decide together with my teams there on what we need to do. Translating science to stakeholders, marketers, customers, investors, become an unexpected skill. Microbiology trained me to synthesize complexity into clarity, and that has been a crucial skill in the boardroom. I now lead teams that sit at the intersection of science and technology, And I'll be honest, the boardroom can feel more foreign than the biosafety cabinet. As a scientist, you are trained to be cautious, evidence-based, and methodical. But leadership demands decisiveness and amid uncertainty, the ability to rally people before all the data is in. It was uncomfortable at first, but I realized the scientific mindset, hypothesis, testing, iteration is actually a powerful leadership model. Microbiology also teaches you ethics. You learn the importance of controls, of transparency, of not contaminating your results. That integrity is essential in leadership. People must be able to trust you, even when the results are messy. And so it was the same adoption of this that I now say I'm not a former microbiologist. I know I'm actually a microbiologist who works in business. That lens still guides how I make decisions, frame problems, and build solutions. Whether it's tackling antimicrobial resistance, sustainability, or food security, microbiologists belong in a room. Indeed, during the COVID years, it was leveraging on the microbiology principles of risk assessment and containment that as a company, we were able to function quite normally and continue serving our customers. Due to our ability to protect our staff and yet carry out our operations, our business actually grew during the COVID years. Credit must also go to the Singapore government for their open and regular communication, practical and effective policies shaped by advice from experts here and abroad that ensured the livelihoods of most people were protected and the nation came through the crisis largely unscathed. Finally, to students and young scientists here, don't let anyone tell you that your path must be linear. Follow your curiosity. Learn to communicate your science. Seek opportunities to apply what you know in new areas. To faculty and mentors, thank you for training not just researchers, but thinkers, problem solvers, and future leaders. As we celebrate 100 years of microbiology at US, I feel an immense sense of gratitude to this department, this discipline, and this community. You have given me a foundation that I've built a life on. Let's continue bridging the gap between bench and boardroom, between science and society. The world needs microbiologists, not just in labs, but in leadership, helping to solve the great challenges of our time. And so here's to the next century of discovery, innovation, and impact. Thank you, and may we all keep cultivating, growing, and leading, both in culture plates and in life. Thank you.
Thank you. Thank you so much for this inspiring presentation, Dr. Tan. Do we have any question to Dr. Tan? Yes, we do. No? Maybe I have one, a provocative one as a microbiologist. Have you seen over time resistance when you keep feeding these animals with your molecule? Do you see resistance developing?
Well, I think what we have observed so far in the field, we've applied this product now for close to 20 years. And today, more than 500 metric tons of feed around the world are treated with this product. I have to say that they continue to be effective, and the best way that we know is when we have repeat business. That means the same customer came back to buy from us, and they continue to raise their flocks, whether it is birds or pigs. And I have no time to show you that now it's found its way also in the application in cattle. And in the United States, at least, you are now able to purchase this over the counter for traveler's diarrhea.
Okay. Yes, Malik?
Yeah, just a quick question. First, I mean, congratulations on, a fantastically useful product. If it can reduce the use of antibiotics in livestock, it's a huge achievement. But you did mention that, I don't know whether it's in vitro, you said your compound also has antiviral effects.
Yes.
And so have you looked to see whether, for example, in the chickens or the pigs that are given these, do you reduce the incidence of avian flu, swine flu, and maybe other things like coronaviruses, infectious bronchitis virus in chicken, etc.?
Thank you for a very interesting question, Prof. In fact, as I sat there listening to your talk, the same thought came to my mind. And I make a note that I must get in touch with you because subsequent to all this, currently we are now putting together a big research proposal on the use of microorganisms as for exactly the same thing you talk about, which is antiviral. Until now in the industry, antivirus there's a paucity of antivirus. And so I think that we have some ideas. Perhaps we can share with you and talk to you and see how we can collaborate in this area, because we do need experts, in virus species such as the H5N1 and the like. Yes.
This is great. So who said that virologists and bacteriologists could not work together, right?
No.
Okay, so thank you very much, Dr. Tan. Thank you. I think it's time to close the morning session. It's coffee break. We'd like to thank again our two fantastic speakers this morning. And I think over to you, Chinwen.
Yeah. So before you go for your break, I just want to do some housekeeping. So there are tea breaks set up right outside this auditorium, as well as MD6 foyer, where the students and the researchers' posters are. So please feel free to join the queue for the tea break. And please help to support our sponsors for today. So they are at the posters as well. So I'll leave you guys to go for a tea break. Please come back before 11 a.m. where we'll start the next session.
Hi, good morning, ladies and gentlemen. So it's my great pleasure and truly an honor to introduce our next speaker, Professor Taniguchi. So I believe that most of us have learned Tada's very critical discovery from our immunology textbook. And Tada is currently a professor emeritus in the University of Tokyo. And he is the foreign member of the Academy of Sciences, USA, and the editor of the PNS, as well as the senior editor of the eLife. So without further ado, please join me, welcome Professor Taniguchi for his lecture.
Thank you very much. It is such a great honor and pleasure to be invited. So this kind of, the most memorable event celebrating 100 years of microbiology and immunology. I thank all and I commend all those people who really made such a great endeavor, including, of course, past and present directors of these departments. And I also thank Yang for kindly this kind invitation. I'm also very happy and proud that there are quite some of the former students in my lab, and most typically Dr. Rosemary Tang in the audience. And she did very well in our lab, and they became a very successful scientist. And she's not only successful scientist, but she also has multiple talents. So she's running many of the venture companies now, and all of them stand to be very successful. I'm very happy to see that. Now I learn more from her than I'm teaching. Anyway, so this is the title of my presentation today. And the topic of my talk today would be first about the classical view of molecular biology of the cytokines with immunomodulators, as you know. And then go into further details on regulation of type 1 interferon genes. Then for some reasons, we encountered in these self-derived molecules released by cellular necrosis. And so those are called damage-associated molecular pattern, DAMPs. And in the context, particularly I talk today in the context of tumor microenvironment regulation. Now, the last part, I spent some time, which I prepared yesterday and in the past 2 days in Singapore, about where are we going, but I think it's very provocative, but I'll be happy to discuss with you, although I'm not a very specialize in this field. But anyway, nevertheless, I think it's a very important issue we have to think about. So when I came back to Tokyo from Zurich, where I did my PhD study, cytokine again gained much attention because they do modulate the immune system, as you know, very well nowadays. But at that time, it was the typical feature of the cytokine was that given cell produce multiple cytokines upon stimulation, either by virus or by antigens or whatever. And then they are produced simultaneously. So it's a mixture of cytokines in this laboratory. And furthermore, there was a functional redundancy and pleiotropy of cytokines has been known. So given biological function is often mediated by more than one cytokine. So it is difficult to elucidate what a single cytokine molecule is doing. So in this context, interferon was getting much attention in those years, the days, this is Time magazine. And so really the approach at that time was the use of recombinant DNA technology, which was just in the beginning, and isolate the cDNA or make cDNA library and isolate the gene encoding cytokine. Once you have that cytokine gene, then one can produce it a single molecule in, for example, in E. coli or cultured mammalian cells. And furthermore, the gene isolation makes it possible to elucidate the primary structure of cytokine. Nothing was known at that time. And and then if you can study also regulatory mechanism of cytokine gene expression for each cytokine gene. And so once recombinant interferon is cytokine is produced in a single form, we can now also analyze biological activity of this particular cytokine. And the clinical application, of course, was much there was an aspiration for the clinical application of the cytokine. And of course, signaling pathway study was also possible with a pure form of cytokine. Now, so in 2016 English journal that the cytokine is important in advanced artificial world nowadays. And so anyway, it's a very attractive molecule at that time. And as you know, there are several types of interferon. This is a very simplified view, but nevertheless, typically virus infection inducing expression of interferon alpha and beta are called type 1 interferon. On the other hand, antigen stimulation of T cells typically produce interferon gamma, the structure of which is very different from type 1 interferons, but the common function they have is impart antiviral state on target cells. Now, so this is the first paper I published in 1979 about the cloning of interferon gene. It is perhaps the beginning of the cytokine molecular biology. And then I went to Harvard to work with McCluskey and other people who became very famous afterwards in other fields. And then here, the typical assay, perhaps young people, it doesn't matter nowadays, perhaps, but nevertheless, at that time, we are doing cell protection assay. So this is a wells, and then no extract, no virus. The cells get of course remain the same, they are alive, and with extract, then no extract, no virus infection cause cell death, of course, and they kill the cells. And here, E. coli supernatant can protect the E. coli supernatant of E. coli containing this plasmid, which we expected to be producing interferon. We is protected and neutralized by this anti-fibroblasts or beta interferon. at that time. Beta interferon antibody completely neutralized. The leukocyte alpha interferon antibody didn't do it. So this way we were able to produce it. And similar work was done in my lab. Actually, I did the work and interleukin 2, another cytokine, class of cytokine. And those cytokines, as you know, it's being used for hepatitis treatment, and nowadays still used for interferon beta is used for multiple sclerosis. And IL-2 can be utilized for anti-tumor immunity. In particular, in these days, it's very important for CAR T cell therapy because we have to grow T cells in vitro by IL-2. Now, we also, this is Charles Weissman is my mentor at the University of Zurich, whose group was cloning interferon alpha genes. And one day, when we finished cloning a sequencing, we stuck together and found that these two genes are structurally related. So this is perhaps the first cytokine, the gene family identification. Of course, there are many cytokines that have gene families, but the interferon is one of them, as I had given earlier. And so now we went on to study regulation mechanism of type 1 interferon genes. And so what is the induction mechanism? This is a very simplified view that the virus infection usually causes induction of transcription has been thought at that time, and it is true nowadays, of course. Then transcriptional regulation makes this interferon messenger RNA inducible, and subsequently interferons are produced to impart antiviral state on target cells in general. Now, Shee Odo, a very talented postdoc who joined my lab, he took 5' franking sequence of the interferon beta gene and linked to the herpes hamilton kinase sequences, and then transduced into mammalian cells, and upon infection by viruses, we saw that this hamilton kinase is virus-inducible, messenger RNA is virus-inducible. That means that the 5' franking region, non-coding region, contains regulatory elements which respond to virus infection and activate transcription. Now, afterwards, it turned out that this element is PD1, PD3 designated by Tom Manakis and colleagues. Anyway, these sequences, which we found appreciated in my lab, found to be very important that they are virus-induced. Then, for the interferon beta case, there is NN kappa B site and there's ATFC gene site in the 5' region, a regulatory region, so to say. Interestingly, on the other hand, interferon alpha genes, they don't contain so many transcription factors, but they do contain the element which is very similar to interferon beta gene at the promoter region. Yes. So then I moved to Osaka University, and afterwards my very talented graduate student, Masashi Miyamoto is now professor of Kobe University, and did a brave approach that the phase screening of cDNA library, so that it's expression cloning, so that some phase containing cDNA may produce the protein which binds to the cis element of interferon beta promoter, which we identify. And so he was able to look for that way. Then this piece of P32 labeled probe, a positive clone, you can see it looks pretty messy, but he's a very careful man, and then we ended up isolating a very, what can I say, a very clear picture. And this clone indeed contains a transcription factor, which we named IRF1 cDNA. So that's the beginning of the IRF family. Subsequently, Hisashi Harada, whom I mentioned before, he used IL-1 cDNA as a probe to screen related genes and he found second factors, transcription factor, which we call IL-2. IL-2 is, anyway, I don't go into the detail of each function of these IRFs. And then, thanks to other people, that other IRF-related transcription factors have been discovered. Now it contains in 9 members, which each contain DNA binding domain on N-terminus region and variety of regulatory regions, so to speak. Now, Mitsuharu Sato, who is working in Singapore together with Rosemary, he found, and in RF, he found that among those IRFs, IL-3 and IL-7 transcription factors behave very interesting. The behavior is interesting. Now, so this is uninfected cells. Then this transcription factor, you can detect it in cytosol exclusively. But after Newcastle disease virus infection, they undergo nuclear translocation. And the same is true for IL-7. So this prompted us to think that those two factors are very important in virus-induced activation of this gene. And similar observation was made by other more or less simultaneously, I would say. And the final evidence we will for the importance of this transcription factor done by gene knockout studies. And again, Sato and colleagues in was knocked out IL-3 gene. So when IL-3 gene is missing, the interference induction of alpha and beta is abolished, but not completely abolished. It's greatly diminished, indicating the role of IL-3 in the induction of interferon gene. And then Kenya Honda is a very famous now, the Keio University professor. He's very famous for microbiota regulation of immunity nowadays. But he and his colleagues generated IL-7 knockout mice, and we have many data, but I just skipped this. But just to say that if you delete both IL-3 and IL-7, there's no induction by by virus. And with other virus, we found to make a similar observation. And herpes virus and here, there's no immune induction. So these two IL-3 and IL-7 is essential for gene regulation. So then, after we have made these achievements, there's the discovery of toll-like receptors. And so now with toll-like receptor discovery, and then it's followed by cytosolic kinase receptors, and made it clearer, I would say, the picture of what kind of signal transduction from outside of the virus, for example, can activate the interferon gene via this IL-3, IL-7 transcription factor. And this is a very simplified view, but typically, toll-like receptors, many of them, not all, but utilized via mediated adapter signaling pathway to have the kinases in the so-called that phosphorylate transcription factors. In this case, cytosolic receptors, another kinase, TBK1 by epsilon, they modify IL-3 and perhaps to some extent IL-7, so as to ensure the gene induction of type 1 interferon. And Takaoka, who is in Hokkaido National University professor, is discovered related to discovery that IL-5, on the other hand, is another member, is very crucial to induce pro-inflammatory cytokines such as IL-6 and TNF, and working together with NF-kappa B. So each IL has distinct functions in terms of the innate signaling mechanism, and this is further illustrate much more complicated than this, but I will simplify this. TOL4 by bacterial derived LPS activation of TOL4 possibly are not quite not very strongly, but via this CBTM pathway, the IL-3, to activate interferon. So 3 reside in the cytosol, and so 7, 9, this recognized RNA, this recognized hypomethylated DNA. They have also now that is through activation of these downstream pathways, they activate IL-7. And in the case of cytosolic receptors, CGAS and others, and STING, those pathways, and RNA-like receptor pathway, which is recognize DNA, this part RNA, this part by DNA, combines to activate this transcription factor. Now, I would like to point one thing that Hideki and I, we now will become a professor, full professor now. They discovered originally that all immunogenic nucleic acid, be it RNA or DNA, they must find high mobility group box protein, HMGB, which reside in nucleus. And we don't know the reason why, but so immunogenic nucleic acid needs to bind and before they deliver to cytosolic receptor or toll-like receptor. And in fact, if you delete HMG genes, the gene induction is gone. Completely. So we don't know the mechanism, but it's an interesting discovery found by Yanai and colleagues. Now, since HMGB is one of the best-known molecules called DAMPs, damage-associated molecular pattern. In other words, dead cells release damage-associated molecular patterns, molecules, and HMGB is one of them. So we wondered whether DAMP plays any role, and so the project was went to 12 direction led by Yanai. So here is the basic principle of immune responses. As you know, normally the immune cells or any other cells in this world, let's say for T cells and B cells, they do recognize intrinsically the self molecule, particularly for T cells, you know, it's essential for positive selection in the thymus. So, but in the periphery, this signal is very low, so we usually ignore this, although it may be important for maintenance of lymphocytes, but I'll be going to details. Now, when pathogen comes in, they release non-self molecules and make strong activation for adaptive and innate immune system can be activated. This is what we call immune response in general, simplistic term. But on the other hand, we note that when cells are dead, particularly by necrosis, they release variety of molecules, which, as I said, it's called DAMPs. And some of those molecules, if not all, activate whatever the receptor, innate receptor in particular. And then response is not as strong as this pathogen infected cells, and but not stronger than tonic signaling, so to speak. So if this is sustained, it could be the cause for accelerated diseases. That's the idea. So we identified several DAMPs, but in particular, I introduce you to some of these, one interesting DAMP molecule. And as you know, massive tumor cell death occurs during tumor growth. Or what you can expect it because they undergo mutation and for selection advantage of the tumor cells, which grow better than the original one. On the other hand, you have to pay the price. That is, when some deleterious the mutations are introduced, those cells undergo cell death, particularly in the context of perhaps necrosis. And you can see tunnel method that massive cell death occurs during the growth of tumor. And there is a report, interestingly, that there's a relationship between tumor necrosis and prognosis. So high necrosis occurs, more advanced disease. Tumor progress more when it is high necrosis. And so this means, doesn't prove, but this means that tumor cell death, although tumor cells die by mutation perhaps, or whatever, hypoxia, these cells release molecules in favor of tumor growth, living tumor cells to grow. If you look at the tumor cells or in surrounding region, there is a structure called tumor immune microenvironment. And usually this is in favor of tumor growth. In other words, immune system is suppressed in this region, microenvironment. So that's what we call cradle of cancer. It's very comfortable cancer cells and they grow more easily. But so why is it so? There are mechanisms, the immune cells which suppress immune responses against the tumor. It could be regulatory T cells and tumor-associated neutrophils and myeloid-derived suppressor cells, MDS3s. We'll come back to this later. And macrophages also play a role in suppressing tumor environments and production of those cytokines which suppress immune response is also very critical. And at the end, this microenvironment, anti-tumor immune response is inhibited. That's why it's called the cradle of cancer. Now, Shee Hangai and Hideyoki Yanai. Now, Shee is now in Harvard, and maybe he's coming back soon, and because he did very well. And so when those people studied and identified an interesting protein as a new dump, that's called TCTP. TCTP stands for translationally controlled tumor protein, TCTP. It's a small protein, 23 kilodalton, and the function of this is not very well studied, but this protein seems to be dispensable in the mice because knockout mice shows normal growth. But of course, who knows the physiological function. But in the context of DANT, this molecule is interesting because, anyway, I'll go further, explain further later. And the expression of TCTP proteins increase in human colorectal cancer. And here, the amplification of TCTP gene they correlate with the poor prognosis. And so it means that TCTP is doing something bad to our body for tumor to grow. Now, in vitro studies, they prepare recombinant TCTP protein, and they found that it strongly induces the chemokine genes such as CXCL1, CXCL2, and even TNF and IL-1 beta. So inflammatory cytokines, they do produce, induce. So it's quite interesting. Like it shows TCTP is inducing this kind of chemokine. And why it is interesting? We found that TCTP transmits signals via binding to toll-like receptor, 2 and 6 homodimer, heterodimer complex. So if you take TOL2 knockout mice, macrophages, TOL6 macrophages, and it's stimulated with TCTP in vitro, there's no induction of those chemokine genes whatsoever. So this protein, indeed, self-derived protein, when exposed outside of cells, they activate innate receptors clearly. Now, we asked whether it is extracellular TCTP and for the promotion of tumor growth, it is our conjecture, but we have to prove this, and so we produce a colorectal cancer cell line which secrete SL4 cells lacking endogenous TCTP by CRISPR-Cas9, and then introduce the cDNA which express CTC with IL-2 signal sequence so that the protein gets all secreted outside those cells. And endogenous TCTP is knocked out. Those tumor cells also grow very well. Indeed, in other words, it is secreted through extracellular TCTP. Perhaps in the context of tumor growth, it's dead cell-derived TCTP, which accounts for the induction of these peptides and peptides. Now, and then after consistently, I would say, when we study in vivo growth retardation of TCTP cancer cells, if you lack TCTP gene, just make sure that you have different cell line established by CRISPR-Cas9 and knock out TCTP gene. The tumors, in vitro tumor growth, there's no difference. They grow both well in vitro. But when you look at in vivo, the growth is very retarded for this kind of tumor. Essentially, the same observation was made with B16 melanoma and SA cells. So this is consistent with the idea, it's not that proven, that TCTP is important for the tumor growth. And so what happened was, in tumor microenvironment? So you can see in the TCTP knockout cells, the polymorphonuclear MDSCs, suppressor MDSCs are very much induced. And in the absence of TCTP in tumor cells, it doesn't happen. And the other similar observation, most notably this PMM MDSC. And you can also see this structure analysis of polymorphonuclear MDSC, strong suppressor of anti-tumor immune response is very much reduced in the absence of TCTP. In other words, TCTP is doing important things for tumor by recruiting this specific type of immune suppressor cells. And now, this result indicate the scenario that is TCTP promote the recruitment of PMM-MDSCs and suppress anti-tumor immune response. Now, indeed, if you analyze tumor infiltrating immune cells in the TCTP knockout tumor, and as you can see, in the absence of TCTP, CD8 T cells augmented compared to control and toid NK cells, and then on activation markers and so on. So in the TME of TCTP KO tumor, the proportion of CD8 T cells increased, and the activation markers of NK cells are upregulated. And these results suggest that the TCTP promotes the recruitment of these suppressor cells in tumor microenvironment and consequently suppress tumor growth. Now, mechanisms seek insights of TCTP-mediated suppression. As you may remember, the TCTP strongly induced in vitro CXCL1 messenger RNA, and in the absence of TCTP, it's enzyme induction that occur. So it's consistent with the previous data that they activate TCTP activates the stem cell induction via TOL6 heterodimer complex forming. So this is just a summary. Summary is the following. So tumor cell death occurs even in vivo, and TCTP acts on SOL2, SOL6 heterodimers, And for vaccine mainly on MMDSC, we have good data for the protein I skipped. Then these when they induce these chemicals include PMMDSC and then suppress CD8 and NKT. So it's the mechanism is quite indirect, but it's different from CTL4 or MDSC, PD1 and checkpoint molecule into the bigger directly. It becomes this lymphocyte. Rather, it suppress the recruitment of this suppressor. Now, of course, you can imagine, so only the question is whether TCP blocking suppress tumor. We expect this, and here we made the TCP antibody, monoclonal antibody, initially 55A3, and as you can see here, the tumor suppression occurs by anti-TCP antibody. And if you combine it together with anti-PD1 antibody, the effect is thrown out. So then we have made, spent some time to make better antibody, now so tentatively called HY. And this antibody, compared to the classical one, which I introduced you just a few weeks ago, it is very strong activity, and binding affinity to TCP is about 100 times higher. And to that extent, it has the infusion very quickly strong. Now we're working, of course, question is whether in combination with checkpoint antibody, whether this antibody could be clinically useful to suppress for the treatment of colorectal cancer. We don't know, but we are working on this nowadays. Now I spent some time within my given time. This is the interesting picture, perhaps depicting what Albert Camus, the Algerian Nobel Prize winner, writer, said in his book, Myth of Sisyphus, the philosopher. So the man, the Sisyphus is punished by God. And so punishment is the following. He has to roll up this big stone on the top of the mountain. As soon as he reaches to the top of the mountain, the stone falls down again. So what he has to do is go down the mountain and then start rolling the stone again on top of it. He has to repeat endlessly. This is a punishment of God. That's quite intriguing to me. I don't know what you think about what this is, what I see. We are scientists. We climb for the top of the mountain, and as soon as we reach to the top of the mountain, we can see another mountain, top of the mountain. As soon as we go to the second mountain, we can see another mountain. And so, importantly, one cannot see the top of the second mountain or the third mountain top without reaching the top of the first mountain. And I don't know what you think. It's my view, but our science is like this. We are endless endeavor. We have some objective, and once we achieve objective, we can find something unknown. This is a endless continuation, which resembles or reminiscent, I would say, of what Albert Camus said. But this is an interesting way of looking at our life. I'm not saying negatively about our life, it's wonderful, but in reality, why we do science, it's really important. And it's important to look at the top of the mountain. Higher, the better. the struggle itself towards the height is enough to kill man's heart. I don't. This is what it's very similar. So, the last few minutes, do I see there's some saying? Only one, 2 minutes. Okay, so let's go quickly. Well, when we are human beings, and we are science in profession, as a profession, but we have to think about more even the betterment of the world. And so science and scientists are facing with a very difficult time in these days. One could blame what's going on in the United States, but also we have many other problems as well. So, Vicky, I skip this. Now, this is a famous picture, as many of you might know, in Boston Museum, the Paul Gauguin's painting. Where do we come from? Where are we? Where are we going? Oh, so where did we come from? This was Japan, based in Singapore. Anyway, so you can laugh at me, but anyway. So Earth was beginning of the Earth is somehow 50 billion years ago, and the birth of life occurred about 30 billion. And dinosaurs, the Jurassic Park world occurred in 200 million years ago, and then we are here, present. Now, through dinosaurs, we have birth of humanity, maybe 7 million years ago, and then it is present here. Then, birth of humanity, and Homo sapiens appeared in the meantime of 0.3 million years ago, and in the meantime, after Homo sapiens, we are crossbreeding with Neanderthals, except for those who stayed in Africa. And then 10,000 years ago, that's important. 10,000 years ago, agriculture and livestock farming started. So I'm not saying something wrong, but anyway, this made our life easier. You don't have to go hunting. You don't have to go to find the rice somewhere. You can do it by yourself. But we can say that this is the beginning of destroying of our Earth if we don't do it, not well controlled. Now we go to this agriculture issue. Then, soon later, BC 500, Archimedes or Lao Tzu, the famous, I love this guy, philosopher of China, and Socrates is here. And then we can see Industrial Revolution, here in Great Britain, and Energy Revolution. Now, then at that time, people like Mozart and Beethoven and Immanuel Kant appeared, among many others, which is biased, so can say this thing for me. And in about this 1900, Friedrich Nietzsche and Igor Stravinsky appeared as two. Now, at present, we can see DNA structure resolved, recombinant DNA technology available, e-medicine, and AI. Now, if you look at the entire thing, from the Homo sapiens to present, everything is portrayted here. A very, very short time window in terms of this big evolution time. Industrial Revolution, Beethoven, e-medicine, AI, this occurred very, very recently, altogether. Now, where are we going? I will draw your attention in context of the planetary boundary, the tentative assessment of the safe limits of beyond Asia on 9 critical properties. And I don't have time to explain all of these, but the climate change, to be familiar, we are in serious situation. Tokyo is 36, 30 degree Oxford nowadays, and then there was fresh water. And one example I show you, also this is the planetary boundary water. So 9 elements in here. So already we are 6, we are also already out of control. 6 items. One example, this one. the latest edition of Real Climate Report. And the average change in population size is more than 5,000 vertebrates, which shows a decline of 70% of the vertebrates disappeared from us. We don't care because we would have familiar animals around it. It is very, very serious because it's ultimately greatly affects our economy or whatever it is. I'll get that time to explain. So anyway, we have to do something urgent, and it is only human being who can do something for this betterment of us. And so this is famous my great alumni of Zurich, University of Zurich. Einstein said, "You cannot despair humanity since we all ourselves are human beings." But we hope that we have to take this point also into integration, into the point, and then for the betterment of the world. And science has to continue, but nevertheless, we shouldn't forget about this aspect. Thank you again, and congratulations again for the 100 years anniversary of the Department of Microbiology and Immunology. Thank you very much.
Thank you very much, Tara, for your wonderful talk. But due to the tight schedule, we probably will move your discussion to the lunchtime. Thank you very much. So our next speaker is Professor Lisa from the ESTA. So Lisa currently is the executive director at the ESTA Infectious Disease Lab and holds a joint appointment at the Medical Research Council, ESTA. So she mainly focuses on immune responses to the epidemic and endemic viruses such as the chikungunya, dengue, as well as SARS-CoV-2. So due to her significant and a lot of contribution to the field, she received numerous awards, including the most inspiring woman at the Great Woman of Our Time Awards and the National COVID-19 Awards, as well as recognized as the SNAS Fellow. So Lisa, floor is yours.
Thank you, Yung Liang, and it's really a privilege to be here this morning to give this talk. So again, congratulations, Kevin, and also to other members of the microbiology department. So this morning, I would like to share a story in my science journey in arboviruses, and that it always holds a special place in my heart because a lot of the work that I'm going to be talking about today was actually done by the PhD students which from this department. So of course, I don't need to introduce very much about the various problems that infectious diseases have caused. Right? I'm not going to give you a COVID talk. I think we had a lot of those over the past few years. But this is just to show that why infectious diseases continue to play an important role, which I think many of the faculty members here, the scientists and the students actually are continuing to work on. Of course, it's not just about respiratory diseases, but antimicrobial resistance is another area that I think a lot of focus should be put into. And of course, the vector-borne diseases, which we'll see a bit later on. Just a little bit of timeline, Professor Malik actually gave the story on the H5 influenza. So I'll just give a little bit of a Singapore flavor. And incidentally, you see that it actually started, by my time, it started after 2000, because I think life before that was a blur. Can't really remember what was happening. But I have to say that I think a lot of exciting episodes of outbreaks actually happened, which some of which sort of changed my life and also my perspective to research. You can see that the chikungunya outbreak in Singapore from 2007 onwards, and then after that we had various outbreaks of flu and also Ebola. That of course Singapore was not affected, but then there was a Zika outbreak in 2016, and I think the COVID took away a lot of our energy. Of course, there's the Mpox scare 3 years ago, and even this year. And of course, Professor Malik has talked about bushfire attacks. Just a little bit of introduction to the A-Star Infectious Diseases Labs, or AIDL, which I'm currently the executive director. And we actually work very closely with many of the members here at the NUS and also the NUS, but LKC and NCID. So we have different three research core pillars: vector-borne, antimicrobial resistance, and also respiratory diseases, which aim to put together various cross-cutting capabilities that answer a lot of important questions out there, from diagnostics, prognostics, surveillance to therapeutics. And this is the current core research faculty. We have both new members and older members, and also younger members that we hope that will assume leadership positions eventually. Of course, our newest addition is Steve Baker, who moved from the Cambridge University just 2 months ago. And I think also many of you are working with various members here in the list. So what we do, it's a lot of basic research, answering key questions from pathogen biology, disease mechanisms, pathogen evolution, and also the various infection models from bacterial, viral, fungal, and parasite. And how some of these questions would then be translatable to the various outcomes here in terms of therapeutics and diagnostics again, and also some of the vaccine designs. And this is just a encyclopedia that all the various expertise from the 12 faculty members, the various vector-borne pathogens from alpha viruses, the viruses, and also Plasmodium, and the AMR pathogens, specifically the escape bacteria, and also some of the Candida species, and also the respiratory pathogens that covers coronaviruses, mycobacterium, and also influenza. And of course, the other pox viruses, such as displayed here, and the ED71, which was a collaboration that was done together with Sylvie, and of course, some of the small projects that are dealing with the fish viruses. So I think coming to alpha viruses, again, it is where it will always hold a special place in my heart, especially for chikungunya. So specifically, so, in a post-COVID world, there was an article from the BBC that indicated as the climate change and mosquito-borne viral infections actually will continue to cause major concerns worldwide, and we will see why, because of the current ongoing outbreaks in Sri Lanka. So in terms of mosquito-borne diseases, I think for those who have been working in dengue and also chikungunya and some of the other related mosquito-borne diseases, I think we know the importance and continuously, despite availability of antivirals and so proper clinical care, we still have quite a lot worldwide come together. There are a lot of cases continue to be a problem. So specifically in chikungunya, right, I would say that back in 2007, when I first started my group, A-Star, and then some of you may know me, my previous life on the Singapore Immunology Network. So at that time, when the devastating outbreaks happened in the Indian Ocean Islands, I think all we knew then was that it was an alpha virus that was transmitted by the Aedes mosquito. And the typical acute infection would be within a week, with sudden onset of high fever and also rash and painful joint pains, and which some patients actually continue to develop chronic disease till several months after. So at that time, there were no known antivirals. Of course, people were trying some of the other common antivirals around. At that time also, there were no licensed vaccines, which very little knowledge on target cells, and also, of course, the immunopathogenesis of the whole infection. So, of course, clearly, fast forward several years later, then there were different pockets of outbreaks. Right? We had other, what happened was, of course, after the 2006-2007 outbreak, which then spread over to Southeast Asia. Actually, then there was a re-emergence in 2013 over in South America. And then that actually created quite a lot of confusion because the different genotypes of the virus was actually not the same. But I would say it was also an exciting time because a lot of the earlier work that were done on other alpha viruses such as Ross River virus and also some of the earlier work that was done on Chick, it actually gave a lot of a nice opportunity to revisit some of the old questions. So clearly, this also then just shows, the more up-to-date outbreak you can see. You can see that continuously, despite the fact that with very, very good mosquito control, vector control, I think we can see that continuously there will always be sporadic outbreaks that keep happening. So, of course, chikungunya is not the only alpha virus that is a problem. I mean, there are other some closely related ones as well, such as O-myong-myong, and also the Mayaro virus, which I guess some people are working on. That's based really in the South America. So I just want to say that, actually since late last year to early this year, unfortunately, in the 20 years on, we sort of is restarting again. Of course, at this point, I think there are no cases in Singapore yet, but I would say in the Indian Ocean Islands, in Sri Lanka, there have been new cases and throwing out new questions that actually they're not able to answer back then. So I just want to share again, as I mentioned, the various parts of the stories that I'll be talking about. I think I've been working on chikungunya for close to 20 years now. And some of all the questions that were unknown then, and how many of them were actually answered by the PhD students that were being there. And I have to say that I myself and also the students benefited from the various discussions from the various members here, being the TAC members such as Catherine. Over the years, there were other members on this. So we did quite a lot of, the students did quite a lot of interesting things. They looked at epitope-based vaccines, the SNPs, also the importance of toll-like receptors, and of course, in terms of immune cells and cell-cell interaction, we also uncovered the importance of some pathogenic T cells, and also the establishment just pre-COVID. We published a paper on the live attenuated chip vaccine, which was actually pretty exciting at the time. So I would say that the students over the years actually really did a lot of very exciting work. So all these were the PhD students who, of course, they have gone on to further develop their successful career elsewhere. So again, from understand questions that are related to innate immunity and also the various parts of their disease, the inflammatory signals from acute diseases to chronic. Of course, that was done in close collaboration with TTSH and then NCID referred by Prof Médecine, David Lang, and Angeli Chow. And also in terms of some of the early understandings of the cellular mechanisms, then allowed us to further develop some of these into therapeutic interventions against chikungunya and also closely related alpha viruses, again, such as the epitope-based and live attenuated vaccines, and also the different classes of post-directed therapies. So I will just split my talk into two parts, right? So I'll say that in the first part, how some of the unknowns in the cellular mechanisms in those days, and some of the more seminal work that actually allowed us to develop further interesting questions. So at that time, this was one of the earliest reviews that I had put together. It was a question mark all the way, as you can see from here. It was very, very unknown, and a lot of the questions were based on what was known for other alpha viruses, such as Ross River virus and also the Peiris virus or the seminiferous virus, where a lot of work was actually reported using mouse models, which may not actually be that relevant to this. But nonetheless, one common mode of transmission was clear. It was the release of the infectious viruses from the infected Aedes mosquito through the skin, and then how it then go to the blood stage. Of course, some of the questions at that time we sought to answer that over the past 15 to 20 years. And again, so at that time, without the fundamental knowledge of chip, actually there was little tools that we could actually further develop to further study the disease and also help with clinical management. So one of the earlier work, so this was my first PhD student at that time, together with NUH, as it was Paul and Raymond, we were able to provide a series of acute infected patients, and actually through that we were able to identify a live virus that was in the CD14 plus monocytes. And so, this was one of the first demonstration where the monocytes was a key target of alpha virus, because before that, it was actually hard to prove this. And actually through this, of course, with the reports from other groups from France and Sri Lanka, and also the US, this was a hypothesis that potentially, how the virus will actually hitch right into the monocytes and then mature to macrophages, and then they will migrate to the joints where they cause the ongoing things. This was the hypothesis then. But I would say, because of all these, various pieces of work that came together actually allowed more questions and to be addressed. So, of course, I'm not going to go into details for the different parts of the story, but I said with that findings in the CD14 monocyte subset, it then allowed a lot of other questions, allowed us to actually identify the importance of the other type 1 interferon genes, and also the involvement of the toll-like receptor 3, and also some of the other IS genes, which we have also shown to play an important role in driving the whole disease. So along the way, the group with I think there's a lot of work. I think we also managed to develop very relevant human surrogate infection models that actually allow us not just to understand the importance of T cells, CD8 T cells, CD4 T cells, also the various parts of the various cellular factors involved in the immunology pathway. So this model, I think, is fairly useful. I think many labs actually have the narrow runner. So you could see from here that actually the wild type black 6 animals actually will develop this giant swelling. And also the two peaks, as I mentioned, the first peak, the viremia, and then the second peak is the inflammation. So I would say this also enabled a lot of interesting stories over the years, but also allowed us to partner with many people around the world, both in Singapore and elsewhere. So that actually allowed the group and the various teams to generate rich source of reagents that used for detection purposes, even some of the seroepidemiological tracking purposes, not to mention preclinical vaccine design as well. So the current work, I would say that my lab is not that big at this moment, but based continuation of some of the interesting work. It's the work of the current PhD student Vanessa. Again, she's also a student at the London School of Medicine, and I have to say, Lim Yang has been very, very instrumental in giving her very strong a lot of encouragement. So she is actually looking at age. I think this is where we have evolved with times. What other questions can we ask about chikungunya, especially the current outbreak in the Indian Ocean Islands. They actually have reported that I think the elderly are the ones who are suffering quite severely from the disease, and also the very young infants. So I think this was a project that we started about 2 years ago. Okay, there's this thing where she's actually interested to look at chick infection in both younger and older groups. So of course, I'll just show you some unpublished data here where using the same animal model, actually she found that in fact, by comparing between 3 weeks old and 4 weeks old, these are the younger animals, you can actually see that the infection profile and the kinetics are very different. You can see that in both the non-immune subsets and also in some of the other subsets, the younger animals, the fibroblast cells in the younger mice are actually more susceptible to infection. So I think I'll just hope that, maybe in a year's time, she will be able to present here at the department on some of her findings. So I will then move on to the second part where with the earlier part I mentioned about the understanding of the cellular mechanism and all that. So now, how with the relevant human zoonotic infection models also allow us to then put together some of the therapeutic applications from epitope-based vaccines to what can be used to also those directed therapies. So I would have to say that over a period of quite a while, there's a piece of work by our 2 to 3 PhD students at that time, and also in collaboration with actually several other members. What we actually found different was that some of the what was known for alpha virus T cells pathogenesis actually was very different from Shee. So I would say that in other alpha viruses such as Ross River and some of the others, it was actually reported that CD8 T cells were the important ones. But actually when we tried repeatedly, when we compared between when we use the CD4 knockout and CD8 knockout cells, actually we showed that it was the CD4 T cells were the ones. And also over with all that, we were actually able to demonstrate that the specific parts of the pathogenic epitopes within those that were driving the actual key joint inflammation during the peak of inflammation of 5 to 6 days. And then because of that, it actually allowed the team to try out various host-directed therapies to actually control the virus-induced joint inflammation and targeting the T cells. So one of that was the use of various T-cell immunosuppressive drugs. So we tried various, and then there was one particular one, the one that the student was almost going to give up, but this was the one, the FTY73 was the one that was the magic drug. And you could see from here whether is it in a therapeutic approach or the on a different post-infection. It could actually control the virus-induced joint inflammation at the during between 5 to 6 days. And then of course when we looked at the various cellular infiltration in the joints, you could see that it's a great reduction in the CD4 pathogenic T cells. So it was also the time where Brazil and other parts of South America also had large outbreaks of chikungunya. This was sometime in those years. And I have to say that this piece of work received quite a lot of publicity in that case because the clinics actually were considering using this as a potential alternative therapy. Of course, at the same time, when this work was published, Michael Diamond from the Walsh U also published a slightly different study with another monoclonal antibody. So I think it was suggested that maybe some kind of combination therapy would be a way to go to control chikungunya. So then another part of the work, while using the various patient cohorts from TTSH and SLAGI, and also with the mouse models, the team, we also showed the importance of IgG. Now that is not new knowledge. So we all know that most alpha viruses actually trigger very protective antibody responses. But when using the patient cohort, we were able to identify that the IgG3 was the dominant isotype that actually has very strong neutralizing capacity. And also we found that the E2 glycoprotein was actually the key immunodominant viral antigen upon the HB infection. And now with that, we also did some kind of a linear B-cell epitope mapping, which we then showed the specific epitope at the end terminal of the E2 glycoprotein, which we call E2V23, which we then sort of tried this, as a preclinical vaccine. And you can see from the panel on the right, actually all the animals that were vaccinated with the E2EP3 actually developed high levels of neutralizing antibodies, which also had reduced joint swelling upon challenge. And I have to say that there was some interest in this startup UK company sometime during the pandemic, that actually developed this problem. So then there's another part of work that, another piece of work that also we stumbled upon by accident. And actually, this was through a collaboration by Andreas Merritz, whom I think some of you are also collaborating with. Now, he works a lot on a Centennial forest virus, and also it was one of his work where he was doing all these clones in that virus, where some of the mutations, he actually found that it changed the viral kinetics. And so, it was a casual discussion at a meeting. I can't remember which country. And then we were just discussing, well, shall we look at this in the chick? So, okay, within the month, he sent some of the constructs over. And then also, of course, we did some very early infection. And indeed, at the in vitro models, the cell models, we found that the viral kinetics were a little different, but not that exciting. But nonetheless, we then went on to look at it in a mouse model. And I think that was the time where we found something really exciting. Right. So this was a situation where we showed that actually some of these, it actually brought down the geometry monocytes. And so later on, the question was, could this actually function as a potential vaccine? So then, of course, we kept the animals longer. At the end, we sort of did a challenge. And you could see that all the animals were also protected. And I'll say that it was one of the first evidence where we showed that the chick NSP mutation was a viable strategy. Of course, I said, with the success of the COVID vaccines, it sort of reignited a lot of interest in RNA therapeutics. And also, I was actually very excited when I learned of the chick from Villanova, which was actually based on the same concept. Of course, it was a slightly different mutation within the NSGs. So I'll just finish off with the last part of the questions that we are doing now, just a couple of slides more, where the current work that we have been doing, I think we got derailed by COVID, which was the work of my senior scientist, Sylvie, and actually she's looking at the host pathogen vector interactions. And this was actually in collaboration with the DBS and MES. So of course, she's actually quite interested in the effects of some of the mosquito salivary proteins and whether does it enhance the virus infection or does it control the virus infection. So in one of the earlier work, which they are waiting for the news of this paper, it's this salivary peptide X, which I will recall it is cellokinin, right? Actually, we found that it was actually abundantly expressed in the salivary of the female mosquitoes, which actually resembles, is homologous to several human proteins and that behave as a full agonist of human receptors. But in any case, what she found was that during the stimulation of this peptide of cellokinin on the human monocytes, if you remember, it's very important for chickpea. They found that it reduced the activation of the human monocytes by downregulating all the type 1 and 2 genes. And then with that, when we tested this as a therapeutic potential, we actually showed that a treatment of the animals with cellokinin also could control the virus induced joint inflammation by actually controlling the monocyte activation. So it makes sense by looking at the joints, all the pro-inflammatory factors are all downregulated as expected. So could this actually be another proper possible approach actually as a host-directed therapy to control viral arthritis? I think we will need to discover that later. So just to complete, I think I will still continue to look, to bring different kind of approaches together with collaborations from all around, how we understand disease immune dynamics that actually then allow us to further answer some of the specific mechanisms, and also how some of these infection models will be useful for various therapeutics. So I think with that, I would like to thank actually as you work, like I say, it's almost 20 years of work, and also with various people from all over the world, people's colleagues from SIGM, and so this is my current lab now. I think my lab members are quite shy, so we decided to use the Ghibli studio version, and with that, thank you for your attention.
Thanks, Lisa, for the great talk. Maybe one or two questions. Anyone?
Thanks.
So Lisa, So you focus on chikungunya virus. So it looks like, there's a cell paper also, companies there's a live attenuated vaccine is, is there. So what's your opinion about live attenuated vaccine? Because think about it, we already have the mRNA technology, we have, protein substitute vaccine. Why I'm asking you this question? Can you also elaborate a little bit the comments about the vaccine for the SARS-CoV-2? Thank you.
Wow, that's multiple questions within one question. Okay, I think for the chick vaccine, yes, each chick, that's the one that went through clinical trials. That was the one, the live attenuated, that it was also based on some of the mutations or attenuation of the NSP. I have to say that there were some problems with that particular vaccine, in the sense that in the first trial, it was also very limited to the demographics to a group of, it was not the right population. And I think subsequently they attempted to go further on, which I've also again based on some stories was that there were some issues in terms of the protectivity. I mean the protection. So I think that actually opens the door of more possibilities of other kind of vaccines. So I think if you are talking about the COVID vaccines, right? I think you'll be in a better position to discuss that. But I have to say that sometimes I think the thing about vaccines now is are people expecting to be protected totally from infection, or are we looking at a vaccine that will help reduce the disease? So I think this is something that we need to be very clear about. I would say that just based on the E2, EP3 preclinical vaccine, you can see that all the animals, I think it doesn't really prevent full infection, but it will actually prevent the effects caused by the infection, which is the joint swelling, the one that you reduce the joint swelling. So of course that one it didn't go beyond the animals. I would say that so I can't comment too much on that. But the NSP, the attenuator, I think that's the one that actually holds promise, and I actually believe personally that it deserves actually more attention on it, especially with the current outbreaks. Is it a possible therapy? It may not protect everyone from not getting infected, but if you're get said, it will actually prevent all the other joint problems or chronic disease for example.
Okay, thanks Lisa for your wonderful talk.
Okay, so before I release everyone for your lunch, right? So again, just a very quick reminder, you have lunch set up right outside the auditorium and at MD6. And please take this opportunity to interact with our researchers standing by at the posters. So talk to them and discuss about their work. And also very importantly, I heard from our vendors, they have bought tons of gifts. No one has visited them yet. So while stock lasts, visit them, discuss with them, get some promotional items for your lab as well. So I'll see everyone back at 1:45 p.m.
Good afternoon, friends, colleagues, students, ladies and gentlemen. Hope you had a nice lunch and welcome back to this session. We are delighted to have a very distinguished speaker, Professor George Gao Fu. And Professor George Fu is a very well-known virologist. He's currently professor at the Institute of Microbiology at the Chinese Academy of Sciences and the China CDC. He was previously the Director General of the Chinese Center for Disease Control, as well as Vice President for the National Science Foundation of China. And he's published very widely 500 articles with nearly 200,000 citations. And of course, published in very well-respected journals, Nature, Science, and so forth. So today he will talk about COVID-19, the virus and receptor discovery and development of vaccine and antibody. So please welcome Professor George Gao Fu.
Good afternoon, everybody. Thank you for inviting me to be here. It's a great pleasure for me to come back to National University of Singapore here again. My talk will be focused on COVID-19. Since I was in the front line of the COVID-19 battlefield, so I have been experienced so much, including management, politics, social misinformation, plus science. So today I will focus on the science part, what we have done, and especially in my lab. This is the title. I give you a story. So this morning, Dr. Meng gave a very good story on from a bench to the board. Now I give you a story from the basic research to bedside when you have such a pandemic. So in my lab for a long time, I'm working on the basic research. I'm working on how the virus gets into the cell, including the avian flu Malik mentioned this morning. I'm working on many viruses, especially for the undeveloped virus, for the receptor and for the host receptor and virus ligand interaction. At this time, we tried to do some work for the COVID-19 virus. Meanwhile, we tried to develop some therapeutic and preventive agents. I will give a full story about this. This is why I call it the virus discovery, receptor discovery, and also for the therapeutics antibody and the preventable vaccine. So where are we now? We are in the post-COVID. The post-COVID, look at this Chinese word. Chinese word is crisis and chance. It's two Chinese words. 危机. I think that from the very beginning to the end, I think we did a lot of work for the risk. We forgot about chance. And think about the chance we have got. With this chance, we put the mRNA technology to be used for the healthy population. That's a milestone. Don't forget this. Don't just mention, oh, this is the risk. It's a challenge for human beings, but give us the opportunity to use the mRNA technology. Think about it historically. It's always like that. Any pandemic or any virus or bacteria challenge, we always try to get this chance. So this is why I want to use this word. Let me explain what I'm doing in science. This is the cell. The cell could be the antigen-presenting cell, could be any cell. Then you have the virus. Mainly in my lab, we are focusing on the undeveloped virus, either measles, influenza, herpes, corona, pneumonia. So that's example. In the interface, there are a lot of protein ligand interaction. This is what we are working on. Then the virus can get into your cell, and then you have a protein dependent or independent processing of the protein with the peptide that will be presented by your T cell. Then your T cell will have interaction with these infected cells. Professor Tada mentioned this morning, and this is the immune cell, how they interact. Again, in my lab, we are also working on these T cell receptors or NK receptors or the interaction of the protein interaction. Some virus, like even for the MERS, HIV, they could directly affect your immune cells, NK cell, macrophage, or T cells. So my lab, again, focus on two interfaces, this interface and this interface. By studying this interface, give you a good chance. Don't forget, don't just measure the risk. Think about the chance. When you do science for this, and that's provide you a good opportunity for the antibody development, for the immunity design. So this is very important. And also this side provide some important for the TCR-T, CAR-T, and also monoclonal antibodies will give you a good chance for this. So this is why my lab basic research for the interface protein interaction. For the applicable part, we develop the monoclonal antibodies, TCR-T, CAR-T, cellular-based therapy, and also vaccine. So why do we have COVID-19? how did we get here with the COVID-19 pandemic? That's a very good question, but it's very difficult to answer. That's why this morning I asked Lisa about the opinion about the vaccine. So we developed vaccine so quickly, and then the virus moves even much quicker than the human beings. Then we have variants, sub-variants, alpha, beta, gamma, delta. And by then, Greek letter only have 24. It's not enough. WHO stopped. No more naming the variants or sub-variants into the Greek letters because alpha, beta, 24 letters. Now we have, I can't remember the current sub-variants name, but Omicron sub-variants like XBB, XBB.1, XBB.5.1, KP.3, KP.1.3. Okay. Well, I can't remember. If you could tell me what is the current one. So why we get so many viruses, so many variants and sub-variants? People, for someone who doesn't know a lot the knowledge about the virus and the human beings' relations, Dr. Taniguchi mentioned this morning. So how old are we? We are only, something like 40,000 or 200,000 years old, the human being, Homo sapien. The Earth, it's roughly about 4.5 billion years. And the microbes, including the viruses, they are 3.3 billion years. You are talking about, we are 1,000, 10,000. We are so young, but we think we are the host, or we are the king or queen of the Earth. And in fact, it's a virus. So this is why from the very beginning, a lot of discussion about is this event a black swan or grey rhino? A lot of discussion. I was a member of GPMB. The GPMB represents Global Preparedness Monitoring Board. In the 2019 annual report, there claimed we might have a pandemic. We might have an outbreak. We might have an epidemic. The most likely pathogens, either flu or corona. From that sense, it is a grey rhino. But are we ready? We are not ready. From that time, from our annual report in 2019, we claimed, we cannot work together. But then, can we work together? Foresee the future. I think we will have even worse situation. Now everybody close their door. No more globalization. Many people against globalization. So we can't work together. So this is our problem. Let me give you a very brief introduction about what I'm involved for one of the meetings. It's called Event 21. There we have a virus. We have a disease called CAPS. That was in October 18th, 2019, just before COVID. Of course, this also made a conspiracy story. Oh, these guys sitting together, they are in the meeting, they are making a virus, they release. America is not a good place, so they'll move to China. Oh, Wuhan is a very good place, they'll release from Wuhan. A rumor like this. So we have a disease called CAPS, coronavirus associated pulmonary syndrome. Look at the name. It's equally like COVID-19. This disease is not a real disease. It's because we have pandemic exercise in New York. And, I was sitting with this lady. This lady was Biden's chairperson of the National Intelligence Council. So then a lot of stars, George, attended this meeting, and with this lady, they are discussing how to release the virus. That's an epidemic. That's an epidemic. You can get all this. It's in the internet. It's in the social media. So that I want to tell you from the professional perspective, any virus coming out, it's normal because the virus is always there. This is, I decided when I did my DPhil at Oxford, I was in the immunology field. Of course, I did my PhD, we call it DPhil, in Oxford. From then, I already know 1970s work from Malik, Peiris. I read a lot of work, and he and his supervisor, James Porterfield, for the flavivirus. But then I moved to immunology. I'm working on the T cells, all this. I thought I would stay in T cells. When I came back to China in 2004, we had avian flu. And then jumped back to virology. And then thought, okay, that's a good field. Let's stay in virology. Now I'm thinking, I should move out. Can we do something for the virology? A lot of epidemics. Anyway, this is the story tells you, this is what we have. The COVID-19 from the very beginning, we know the virus, and then we have the fake, WHO claimed the fake, and then we have a pandemic. For those who is not familiar with the fake means public health emergency of international concern. So whenever you have some risky viruses or big public health event, the WHO will claim fake event. So from the very beginning in 2019, in December, late December, we found in China, in Wuhan area, you have a PUE, pneumonia of unknown etiology, PUE around. And there, initially, because the hospital would invite the third party, would send their samples to the third party service. So 3 companies, they sequenced the samples from, the patients. They found pieces of the coronavirus. Then you remember in the social media, the end of December, claiming there is SARS is back because of SARS-like virus. But it's not a full genome sequencing. The third company, they only claim it's a SARS-like virus. From the very beginning, we already know it's from a third-party company. And then, the China CDC and the Institute of Virology, China Academy of Sciences, and also Chinese Academy of Medical Sciences, so many scientists groups got in. We did the sequencing. We observed the virus. The virus looks like this beautiful coronavirus. They have a crown on the surface of the virus particle. And then on January 24th, Chen Wang and also Peter Hobby, Fred Healy, we published a paper in the Lancet called A Novel Coronavirus Outbreak, a Global Health Concern. From that, we also, professionally, we think that could be a global concern. But of course, we couldn't predict that would be a pandemic. And then we released the genome sequencing with the GSAT, and then we, so many papers published in so many journals, diagnosis, vaccines, by sharing all this. So data sharing is very important. Although we have problem for the data sharing. A lot of countries now, they just close their data. They don't want to share the data. Tell me the exact numbers of the H5N1 in the US. Tell me the exact number in Hong Kong, in Singapore, or in China. The exact number of COVID-19 last year or this year. We don't have exact numbers. So this is the problem. So in my lab and a lot of other labs, once we have this virus, we did the pseudovirus infection, all this. We did the by-capsule work together with KYUA in Hong Kong University. We found the receptor is the same. We immediately found some virus, some is the same. We also got some monoclonal antibodies against SARS, and we saw there's some differential binding or neutralization from SARS. It's different from SARS, but the same receptor as ACE2. And then we, because I'm running a lab with structural biology, very quickly we saw the structure to show how it binds, very similar to the SARS, binding to the Homo ACE2. Retrospectively, in 2016, June, I was invited by a journal called Trends in Microbiology, write something about coronavirus. Look at the title, 2016. We wrote epidemiology, genetic recombination, and pathogenesis of coronaviruses. There we see it is likely not a matter of if, but when the next recombinant coronavirus will emerge, cause another outbreak in the human population. If anyone working in this field, emerging viruses, especially coronavirus, we know one day we will have a coronavirus. It could cause a pandemic. Let's just count how many homo-infected coronaviruses here. The first one, everybody remembers SARS. The first coronavirus, 1965, is called HCOV, Homo coronavirus 229E. So that's, during the early 1960s in Europe, especially in the Netherlands, they isolated The virus is not isolated, but they know their samples. There are some special virus. It's own virus, but they don't know it's a coronavirus at that time. For a few years, finally in 1964, they found one of the samples they labeled as 229E. 229E is a sample name. And then in 65, under the microscope, electron microscope, they saw it's a coronavirus. So they spent several years. They confirmed the first human-infected coronavirus. That's in 1965. 2 years later, OC43. And then 204, you have AL63, and then you have HK1. Remember, previously, all the virus names are either by the discoverer. If George discovered, it's a George virus. It's John discovered, John, like Tyler's virus. Like all the places, like Ebola, and Hong Kong University, Kaiwa found virus. Kaiwa named this one, Kaiwa University, Hong Kong University #1. This is why it's called HK1. At that time, because that's not a serious problem, no one really remember all the virus name. But no one played the Hong Kong University. But if today, if you name a virus, the National University of Singapore virus, you are in, think about it. We can't name the virus like this anymore. But previously, HK1. So this is what that we were pressed to think about where the virus from, origin of the virus. I came here, someone said, George, are you still working on the origin? Shee asked me this question. I said, I'm working on the virus. I'm not working on the origin anymore. You cannot work on that. If you found the truth, if you couldn't find the truth, what can you say? Or can you really find the origin? Maybe you will never find the origin. I'll give you an example. HK1 was discovered 204. It was a patient from Shenzhen, mainland China, traveled back to Hong Kong. Kaiwa got this patient, isolated the virus named HK1. 204. But remember, this biobank is very, very important. The scientists in Brazil, they have some samples. They have some PUE samples in their freezer. So they got out of this. They found 1995 samples. They have HK1. So HK1 at least was in Brazil as early as 1995. Maybe 1960s, something somewhere. We don't know. But this is why it's a scientific question, but it's very hard to explain to the public or to the politicians. So let's leave some time for the scientists to work on the origin for any virus. Tell me where the virus from, HIV from, where CMV from, where's the EBV from? Can you give me exact places where the virus originates? So this is why it's very difficult. Anyway, then we have MERS and we have COVID-19. It's a 7 known, very serious problem. Let's move on a little bit here. Otherwise, and then you have COVID-19. While we were working so hard for the COVID-19 pandemic, and then 3 sporadic human-infected coronavirus was discovered in Haiti, South Korea, and Malaysia. So in total, we have 10 known human-infected coronaviruses. In 2018, I was asked by the journal Cell after the Zika outbreak. So, George, can you write something about the re-emerging virus? Can you foresee the future? What can we do? That time, 2018, I wrote a paper. It says commentary. I said from AIV, avian flu, to Zika virus, attacks from re-emerging pathogens. There, in my very short abstract, I mentioned, okay, what can we do for the public or for the professor? We can only invest your money for the basic research. That's the first thing you can do. Second, do the surveillance. Nothing else you can do. So surveillance, basic research, surveillance, basic research. So that's the only thing you can do. That's in 2018. So after that, we discovered the virus. The virus was initially, because I experienced everything from the very beginning. We got the samples. We thought, think about HK1, think about the coronavirus. It's very hard to grow the coronavirus. So from the very beginning, we thought maybe it will take a long, long time. By then, within 5 days, the virus grew very well in the viral cell. You put the samples in the viral cell, 5 days later, you take the PCR, co-PCR, the virus grows there. And then under the cryo-EM, not a cryo, just the EM, under electron microscopy, you saw the virus. Beautiful coronavirus. And then, anyway, after that, my lab stepped in, we did the vaccine development. The whole world, we tried very hard. So I want to mention to you. So the vaccine plays a very important role. Now we were attacked by, a lot of people, including the Secretary of the Human and Health Services from the US. They thought, vaccine didn't work. I remind you, 2 words: eradication, elimination. Smallpox was eradicated by the vaccine. Polio was eliminated in most of the countries by vaccine. HPV, well controlled in China by vaccine. That's by protein supplement vaccine. What am I saying about this? So, for the vaccine development for the SARS-CoV-2, we have inactivated vaccines, mostly used in China. Live attenuated, that's the ideal, that's our hope, but no live attenuated be used. But the nature developed a new attenuated vaccine as Omicron. All the Omicron subvariants of the virus, they are becoming so attenuated. And then you have protein subunit vaccine. This is some vaccine developed in my lab, including my lab. And then viral vectors, including influenza vector, adenovirus vector, and a DNA vaccine, mRNA vaccine, and virus-like particle. Basically, those 7 strategies being used for the COVID-19 vaccine. Of course, the 7 strategies can be used for any of the vaccine development of any viruses. So the mechanism for this is the whole virus by chemical inactivation. You have an inactivated vaccine. Or get a spike out. This spike is a trimer. Get one of the trimer. You do the dissection, you see the structural elements, what it looks like. And then you do something like 2P or 6P, whatsoever, try to stabilize this trimer. As everybody knows for the RSV. RSV now you have pre-fusion vaccine. It's developed in the world, being also clinically used. Then you also can do the further dissection of this one. You have a receptor binding domain, and you have receptor binding module, RBM. Then you can put RBD as a dimer or trimer. So this is my lab. We developed RBD dimer vaccine. So this vaccine was developed in collaboration with Zhifei Lancome. So this one, we did this for MERS, COVID-19, SARS, and all this, it works. From the very beginning, when we didn't have these variants or subvariants emerged, you face 3 clinical trials. The vaccine, like efficacy is 81%. It's a very good vaccine. Played a very important role from the very beginning. So this the vaccine looks like. The vaccine design, you have 2 RBD, receptor binding domain. They linked by fusion. We fuse them together. This is antibody. You can see the antibody works well. This is the antibody we developed from our lab. You collaborated with Eli Lilly. It was used widely in Europe and America for a while, in 2021. And once you have the escaped mutants, this one, Omicron. Once you have an Omicron, this antibody not working. This is the cryo-EM structure. You can clearly see, how they bind each other. And we can also, by this design, we can always change the subtype. You can have a prototype with beta. You can have delta, omicron. You can create whatsoever you have. This is a dimer. And then we also tried. We successfully made it the trimer. You can have trimeric one. This trimer will increase the spectrum of the neutralization. That's clearly shows you. So this RBD dimer can be developed as either dimer or trimer. The dimer being developed as a clinically approved vaccine, but the trimer also works very well. This is Sai, Ugur Sai and his wife, they wrote a book recently in the vaccine, mentioned about, RBD vaccine from my lab. And also this vaccine be used in Uzbekistan, like 60 60%, although vaccinated people, they have used this vaccine. Over half of the population in Uzbekistan, they use this vaccine. This is something when our president visited Uzbekistan, he wrote something about Chinese vaccine. It's the vaccine he mentioned. And after that, meanwhile we also try to isolate the monoclonal antibody from the patient. So we isolated one of them. We named it as CB6. So this is CB6. The paper was described in Nature. We did all this work in animals very quickly. By April 2020, we already transferred this monoclonal antibody to Junshi. This is the Junshi. It's the top alliance. This is the Chinese company. And this company licensed to Eli Lilly. Eli Lilly developed it as anti-CB1. That's a good example. If you work very quickly, your basic research in the lab, virus entry, molecular antibody isolation, if you have a good partner, you can get it as a product. So they save millions of lives in Europe and with severe patients in 2021. Of course, then it's escaped. Once you have Omicron, that antibody didn't work anymore. So then we did all this with AI empowered. Can we really change these molecular bodies, escaped molecular bodies, into fully armed molecular bodies by using AI? So I encourage everybody here. I'm working on the virus, virology, immunology, structural biology, cell biology. Now I'm working on AI. Remember, you are never too old. We always get the new technology. Now, my lab, we are working on, can we really modify this molecular body? So what we did, by using all these programs developed by David Baker, who got a Nobel Prize, by series of the tests, blah, blah. And then finally, we got one of the mutations we call it CB64. It works very well. So far, it works for all the mutants. Also, we put this one as a bispecific antibody, as a diabody. It works so well. Meanwhile, I'll mention to you AI. So don't forget AI. For AI, we developed the PD-1 binder, a PD-L1 binder. You use a PD-1 molecular bodies, but you also get some escape. And we want to have a artificially made, homemade molecule design. So we designed, in collaboration with Bruno Correa, we developed the alpha helix. If you want to develop any macromolecule, better use the alpha helix, because alpha helix would give you a very good chance, less immunogenic. So this is one, give you example. This is what we have done at the moment. Meanwhile, we did all this. We did the, type 1 on protein fusion by the basic research. We developed a peptide that can be used, called HR3000, as a inhibitor, as a spray, nasal spray. And based on, we already finished up the phase 2 clinical trial. Because now we do have so many places, we can't do the phase 3. So we are still in the pipeline. We are waiting for the chance to get a phase 3 trial. Phase 2 clinical trial works very well. That's also the basic research. We are working on a virus entry, but we found some peptide would work, and then this peptide was developed as products. So this is how it works. We did this work when we have the SARS, long-term commitment. When we have this for MERS, we did the same thing. It works. And when we have SARS-CoV-2, it works. The peptide we developed, we modified that peptide. It works for all the homo-infected coronaviruses. So at the moment, with peptide oriented company, we are in a free clinical trial. So that's a good example. This is what it looks like from Haiyu. That's a Chinese Shenzhen-based company, and we are working on it together. Meanwhile, we also work on the Chinese herbs. You remember that in Wuhan, you saw some in the social media. They have big containers. They have all these Chinese, and the people take that one. So does that work? Of course, we need science. Get the science involved for this. They have all this, we call it all these different formulas. One of them we found from my lab, we did all this, is they have one of the formula, decoction, they have leucopeptide. This leucopeptide mimic the Paxlovid, works exactly the same mechanism as Paxlovid, working on the 3C protease, working on the 3C protease, binding the same pocket as Paxlovid. So this one you would find from the Chinese herbs. The Chinese herbs, they grow as a plant. The plants, they have streptomyces. Streptomyces are metabolized with this leucopeptide. So obviously, some of the Chinese herbs, they have something. We collaborate with Dr. Huang Wuqi. We also did the other decoction. We found they have 4 key components. Some of them they have antiviral effects. Some of them they have anti-inflammatory effects. Cytokine stop. They would stop cytokine stop. Now, what's the next? 21st century, we ought to have so many fix. Remember, pandemic 2009, polio, Ebola. When you had this Ebola, I spent 2 months in Sierra Leone fighting against Ebola in 2014. Myself, I was there. We were worried the virus might spill over, spread all over the world. Then you have Zika and Ebola again, COVID, monkey. Monkey, the first fake was claimed in 2022. And then this is removed, but in August 14th, WHO claimed again. We are still under fake for monkeypox. Recently, in Sierra Leone, monkeypox is very serious at the moment. So we need a vaccine. So then our lab, again, we don't want to have this 200 proteins with attenuated vaccine like pox virus whatsoever or MVA. So we developed 2 components, only 2 proteins. These 2 proteins, we have A35 and M1. I call it the double A35 double M. I put it as a dam, double A double M, dam. Build up a dam. Build up a dam to prevent from the mpox. So this is the work in my lab. This also, at the moment, we are in pre-IND in China. We are ready for some clinical trials. We are applying for clinical trials. RSV, as mentioned, RSV, you have this prefusion form. This is the GSK, Pfizer, they have the vaccine. They have all this. But when you do the scrutinize the structure of this F protein, again, it's a trimer. Look at this head. The neutralizing epitope is always here. The stronger one, 5, and then the less component one is 1. 1, 2, 3, 4, and 5, and 5. So we try to get all these major epitope to link them together again as a dimer. And only the head domain, this head domain. And it shows structurally, they expose all this. But of course, because we removed epitope 1, epitope 4, we kept 2, 5, 5 very well. They provide very good neutralization. This is another sub-unit vaccine developed for my lab. Now, risk and opportunity. Risk and chance. So don't worry about the virus. We learned the virus. We studied the virus. We should learn from the virus. Let me show you learning from the virus, what we are doing at the moment, what we are cooking at the lab. So, David Baker designed this cage. This cage can be used as a nanoparticle for vaccine development. This is David Baker's. So what in my lab, we found African swine fever virus have a protein called A137R. This only 100 amino acids, but they form a cage like this. They form a beautiful cage. This dodical hydro cage looks like by using this cage, we put either SARS-CoV-2 receptor binding domain or influenza HA. We got a beautiful nano cage. And other EM, look at this. You see the purple one is a cage itself. On the surface, you have the RBD. So this is the vaccine we are developing at the moment. More importantly, can we do the immunology plus adjuvant? Can we because this is a very good kit. We did a screening, we did a global analysis. We tried, maybe this kit is very popular. We tried no any other kit like this one, because you want a kit, you have the virus as a kit itself, but the virus is so compact. There's no place for you to insert any fusages or immunogens. But this one, they have a big hole, give you a chance to put everything. Can we really do something like you have an immunogen, RBD or HA head for flu, plus some macromolecules like interferon alpha, interferon beta, interferon alpha, interferon beta, interleukin 2, whatsoever as an adjuvant? We got it. Look at this one. Again, the blue one is a immunogen. And the cyan one is a macromolecule. Under cryo-EM looks like this one. So learning from the virus is very, very important. This is the only protein used every spine fever virus. Finally, I want to mention to you, because from very beginning, we have a risk and an opportunity in Chinese VG. Don't only think about risk and charge, but the public, now we have a biological vaccine. Now we need a social vaccine. I'm writing a lot of books for a popular science. I'm giving a lot of talks for public understanding of science. So now we have a monkeypox. People say, oh, this monkeypox is created, made up by this guy. You know this guy. So we have problem with the infodemic. That's called the infodemic. Misinformation and disinformation mixing together with the correct information. They cover new information called infodemic. The study is called epidemiology. I named the causative agent is for me. I named it as a euphorus. So I wrote you the China CDC Weekly. When I was Director General of China CDC, I created a journal called China CDC Weekly, like the US MMW. So welcome to visit China and go. I have this Malik with me, right? You saw this sculpture statue. This statue erected in Chinese Academy of Sciences campus. Come and with us, have a look at this one. Thank you very much.
Thank you very much, Professor George Gao Fu, for your fascinating journey in COVID-19 and learning from the viruses. So we welcome questions from the audience.
Yeah, so, again, there's a lot of questions and controversy, as you mentioned, about the origins of the virus. So the Lancet paper that first came out, Zhao Bin's paper, 30% of the cases had no association with the Huanan seafood market. Do you think that that is still true, or could there have been, low-level circulation that was maybe not picked up until the amplifying event?
That's as I said, it's a very quite complicated question because we have no answer. It's very difficult to answer any question for the origins. We had a paper early last year published in Nature. We did all this detailed analysis, even store by store for those, it's an open market, right? And we didn't see any relations, with any store. There's some clue, but we tried so hard. We didn't isolate any virus from any animals. Though some are like Eddie Holmes, Michael, and they thought there must be a raccoon dog. But, we tried. We didn't get any virus from raccoon dog. Let me make it clear, give me a chance to explain my understanding. For SARS, from the very beginning, Yi Guan, and he did the work in 2003, from the very beginning. That paper claiming the virus is derived, the origin is from at least civet cat. And Ian Lipkin, he studied the work claiming the MERS is from camel, dromedary camel. In my opinion, we need to revisit that. Why? First, for the SARS, COVID, civet cat, only that special batch, their the first SARS was discovered in Heyue, Guangdong province. From that special batch of the civet cat, you isolate the virus, you did the sequencing, not any other. Now 20 years, no one ever get a SARS virus from any civet cat outside that area. MERS, think about what Ye Lipkin, I talked to Ye Lipkin. He also told me we need to revisit. So if it could be human transmitted civet cat to camel, not the other way round. So we need to rethink about this. So this one, there's no evidence, any animals or the direct evidence with the Hualan. So I was at the frontier. So I have no clue. We want to know this answer, but we didn't have it. 30% or 35 whatsoever. We have all these numbers, but that's no clear answer. Thank you.
Any other questions? As you pointed out, there's a lot of potential for Chinese herbs to treat viral infections, but it's always a challenge to characterize the active compounds and the mechanisms of action. What's your comment on that?
Because historically, Chinese herb has been used so widely, especially with some combination, that means decoxyl formula. Clearly, look at all these books for 1,000 years, and they have a lot of formulas there. I think we need to get this done like the way we are doing to try, especially with mass spec. Because what we found the new peptide by mass spec. You have mass spec, you have the mixture, and by mass spec help us to identify. I think there must be something. Again, learning from the nature. By then, all these molecules are naturally or man-made. They are natural compounds. These natural compounds, we need to try to identify one by one. Thank you.
Thank you. Any other questions for Gao? If not, thank you very much for your excellent talk.
Thank you.
It's now a great pleasure to introduce our next speaker, a colleague from my department, Professor Veronique Angeli. And Veronique has done a lot of work on lymphatic immunology and has done very interesting work on the molecular mechanisms of lymphedema, which is, of course, a very important clinical problem, for example, in cancer patients. And today she will share with us some of her latest discoveries on this molecule called LIFE-1, which is important in lymphatic immunology. So please welcome Professor Angeli.
Okay, good afternoon. Thank you very much, Vincent, for the kind introduction. And so on this very special day, I decided to give you, a bit of a snapshot of my research journey since I started in this department when I joined the first time in 2006 until now. And also, I think it's an opportunity for me to basically pay tribute to people that really have contributed to this particular journey. And in fact, I would like to, before I go into the science, I would like to start by thanking a few people, and particularly Mike here. Thank you, Mike, for believing in me, in fact, in 2006 when you recruit me in the department, and also all for your support and guidance, particularly in time where I went through a rough time in my life. So this I'm very grateful. Thank you very much, Mike. And I would like also to thank Mary and here, because you supported me during my application for my PNT. and which was successful under your leadership. I also like, of course, to thank Nick, and Kevin for your guidance under your headership. So, indeed, in my lab, we have two major interests, and it's really to understand like how the immune system, in fact, control homeostasis of tissue, and particularly we focus on two main components of the immune system. And today I will share like how indeed lymphatic vessels, often neglected vasculature, they can contribute to the maintained tissue homeostasis and also tissue-resident macrophages. But I will start with the lymphatic vessels. So you may not know, but lymphatics are in fact very important to keep us healthy, to live longer, because so they play a very important first to maintain tissue homeostasis. So by clearing fluid from your tissue, they also are very important to, they're part of the immune system. So they transport like immune cells, protein, antigen, your antigen-dendritic cells. And they, now there's a lot of examples showing how they're important to resolve inflammation. But in the lab, we are very interested about the role of lymphatic vessels in the transport of lipids. So in the gut, the lymphatics are important to absorb lipids. But we were particularly interested and fascinating for all these years about the particular crosstalk between cholesterol and the lymphatic vessels. So cholesterol, as you know, all cells need it to form like a plasma membrane. But however, when, you know, the cholesterol levels are too high, then this will lead to, for example, hypercholesteremia and leads to critical disease such as atherosclerosis. So what we found in particular here is like using mouse that develop hypercholesteremia. We show, in fact, that they have an impaired lymphatic transport and also some, and this impairment in lymphatic transport means that they are not able to drain anymore the fluid from the tissue back to the blood circulation. This was related because of changes in the structure of these lymphatic vessels. On the other hand, we found that, in fact, lymphatic vessels are very important to transport the cholesterol. So showing that in this hand, you have, when you have high cholesterol, this hypercholesteremia leads to impaired lymphatic function. But in this case, we show that in fact, lymphatics are really essential to clear cholesterol from the tissue. So if in a normal situation, you have an efficient lymphatic drainage, this will clear this cholesterol, prevent basically this abnormal accumulation in tissue. But when you, for example, in a mouse where we resect lymphatics, so we cut lymphatics, we can see that we basically have an abnormal accumulation of cholesterol. So of course, this particular crosstalk between cholesterol and lymphatic vessels raise even more a question. And in particular, the first question that we addressed was whether this relationship between cholesterol and lymphatics can be relevant to disease where it's known that you have, you know, cholesterol accumulation. And you probably, you know, atherosclerosis, which is this chronic inflammatory disease of your arteries where you have an accumulation of cholesterol. So the question was, can this, you know, disease is like basically involving an impairment of this lymphatic drainage? And this is what we found that indeed, when in atherosclerosis, basically to prevent atherosclerosis, you need a very healthy and efficient lymphatic drainage. But I want to focus more on this question, where it's like whether in humans, because all what I've shown so far it was in a mouse, but really the question is like whether is it relevant to humans, and to address whether indeed, you know, lymphatics are important in this clearance of cholesterol, we decided to study basically like a condition where there is in humans lymphatic insufficiency, and indeed is to look at lymphoedema. So lymphoedema is basically like a cause because you have like dysfunctional lymphatics. In most of the case, like for example, like 50% of this lymphoedema are related to cancer. So for example, women that have breast cancer or cervical cancer, because of the removal of the tumor, also resection of the tumor and also around the lymphatics, plus combination of radiotherapy and chemotherapy. This will lead down the road basically to this swelling of their limb, which is called secondary lymphedema. So it's a very chronic and progressive debilitating condition, and for which there is no cure at the moment. So I think there is no cure because we still don't know really a lot about the pathogenesis of this disease. So what we know is that, of course, you have fluid accumulation as shown on this picture, but this basically fluid retention leads to inflammation, some changes in the adipose tissue as well, and also at the late stage, really fibrosis. So we collaborate with Prof Tanden Kim here at HGH, who see this patient, and we interrogated basically this biopsy from this patient to address whether lymphatic insufficiency in this patient was also associated with an accumulation of like toxic cholesterol in their tissue. And so, to cut a long story short, like what we basically found is that indeed, accumulation of this toxic cholesterol that we can stain for, like with Philippines, so it's the free cholesterol. And you appreciate that indeed, when over stage, like advanced stage, you can see an accumulation of this free cholesterol, whether in the dermis or in the adipose tissue. And we confirm this by mass spectrometry. So indeed, when you have lymphatic dysfunction, you see this abnormal cholesterol accumulation. And what was interesting when we look at further stage 2 and stage 3, what we observe is also the formation of these foam cells. And we know in the field of atherosclerosis that, in fact, your atherosclerotic plaque are a form of foam cells. So this is when macrophage fibroblasts basically try to clear this cholesterol and engulf it, and then become these foam cells. So like in atherosclerosis, we also found this formation of these foam cells at a later stage of the disease. So here really showing some evidence for the first time that lymphedema is associated with this abnormal cholesterol accumulation. So of course, the next will be, can we remove basically this particular excess cholesterol to reverse basically the disease and maybe so to treat the disease. So how to do that? So we were looking at the literature and we stumbled upon this molecule called cyclodextrin because so cyclodextrin acts like as a cage basically and is known to trap cholesterol and then to basically can dissolve this cholesterol and then it will be excreted by the urine. And in fact, there was some evidence in atherosclerosis that cyclodextrin can promote basically the regression of atherosclerosis. So we use this molecule, and however, we had to establish first a lymphedema model, the preclinical model to test this. And so we established a model that you can see here, recapitulate after resection, surgical resection of the lymphatics. You can see these basically lymphedema as you know, I think the picture are really similar to what I just showed you for human secondary lymphedema. So we really exacerbate swelling compared to the the sham operated limb. And in this model, we were able also to show that you have this accumulation of cholesterol. So using this model, we test whether cyclodextrin treatment could basically improve this lymphedema. And indeed, we show that cyclodextrin can reduce this accumulation of excessive cholesterol, and also some really significant effect on the edema, as well as, in fact, what was really interesting to find is that this treatment, so removing this excess cholesterol, also improve lymphatic function. We have some evidence that because it helped to regenerate, in fact, the lymphatic system. So we have hopefully found here really a way maybe to treat this disease. So just first message is just to show how important this cholesterol, the abnormal accumulation of cholesterol can have an impact on our lymphatic system, and in particular for lymphedema. targeting this cholesterol might be really a promising therapeutic. And we think that these, might be relevant to other diseases that are associated with abnormal cholesterol accumulation, like liver disease, neuronal disease, and so forth. And this is what we'd like to explore further in the future. So I'm going to change gear, and I'd like to share what we've done in terms of the importance of tissue-resident macrophages in regulating tissue homeostasis. So you probably know macrophage because of their role in host defense. And in fact, Elie Mechnikov was the first to really describe the phagocytes of our cells when he looked at starfish and how they respond after injury with a rose thorn. And but since then, I think the past maybe 10 years, there is increasing evidence that macrophages are not only a role in host defense, but also in maintaining basically tissue homeostasis. Can be the brain, the intestine, and so forth. And of course, with that, we know now that they are very heterogeneous cells in terms of origin, in terms of their function, and also they have really tissue-specific or niche-specific function. So there's a lot more to discover on these cells. And we were particularly interested by a subset of these tissue-resident macrophages expressing LIVE-1, so which stands for lymphatic vessel endothelial granular receptor 1. So indeed, as I say, I'm a lymphatic, researcher, and we do a lot of staining with LIVE-1 to look at these beautiful lymphatic vessels. But it was when I was still in a postdoc, in fact, I also observed that there were other cells that were LIVE-1 positive, which were not lymphatic, or lymphatic endothelial cells. And I was wondering, what are these cells? And when the first, technician moved to my lab in Singapore, Angeli, she also made the same observation. And this is where we decided to try to understand what are these cells. And in fact, at the time, there was very few papers showing that these LIVE-1 cells could be expressed by macrophages. And this is how we start to work on these particularly population of tissue macrophages expressing LIVE-1. Now, in fact, they've been discovered in a lot of different tissue. By the time, we didn't know what were their function and where they were particularly their specific niche indeed. So what we found, in fact, is that the LIVE-1 positive macrophages were particularly abundant on the outer layer of the blood vessels, and particularly blood vessels that are covered by smooth muscle cells, so your arteries, like your venules, but not on capillaries. So we went on and to design a mouse model where we could deplete specifically LIVE-1 positive macrophages, so as shown here. And what we found is that when we deplete this macrophage, basically, I just shared the summary of this study. What we found is that in absence of these LIVE-1 positive macrophages that usually are in the outer layer of your artery, we observe an accumulation of collagen. And this is because, in fact, we found that there is a specific crosstalk between these macrophage and the smooth muscle cells, which are known to produce a lot of these extracellular matrix, including collagen. And we found that, in fact, they need to interact through engaging, in fact, LIVE-1, which is expressed by the macrophage, and the ligand of LIVE-1, which is HA. And through this interaction, basically, the macrophage is able to control the production of collagen produced by smooth muscle cells. So here was the first evidence that macrophage, tissue-resident macrophages, can control basically the arteries tone and particularly prevent arterial stiffness. So basically, more rigid, arteries. So of course, showing this homeostatic role of tissue-resident macrophage and these LIVE-1 macrophages prompted us next to wonder whether these macrophages could have an importance in arterial disease, which are associated with, for example, arterial stiffening or accumulation of matrix protein, including collagen. So we went on and to study indeed the role of these LIVE-1 positive macrophages in different disease. And first, we looked at vascular aging. because vascular aging is in fact associated with arterial stiffening, and it's known that in fact arterial stiffening precedes arterial disease, for example, atherosclerosis. So it's really a first sign that you have some changes in your the arteries. So we study mice that are 2 years old and that develop arterial stiffening. So we can also look at the stiffening by measuring pulse wave velocity, and this is what is done in clinic as well. And what we found is that, in fact, during aging, so when these live-on positive macrophages become old, they lose this capacity to control the collagen through interaction with smooth muscle cells. So we can see here, these are the data that I showed you previously. For example, in young mice, the live-on positive macrophages are able to control collagen by smooth muscle cells, but in the old, they are not able to do that. So then we look into, can we rejuvenate these cells? And we use one of the strategies currently investigated, which is basically inhibiting the mTOR pathway, which has been involved in longevity. And we use the inhibitor of mTOR, which is rapamycin. And when we treat mice with rapamycin and isolate the live-on positive macrophages from these mice, we could see that we can now restore their function. So there is hope we can rejuvenate our live-on positive macrophages and hopefully basically prevent aging of our artery. And then we also found that these live-on positive macrophages are important in atherosclerosis. So atherosclerosis, as I mentioned, is a chronic inflammatory disease of arteries. And since these live-on positive macrophages normally in a steady state, they are important to control basically the homeostasis of the arteries. We were wondering maybe atherosclerosis is associated with some dysfunction or changes in the macrophage that could explain basically these contribute to the disease. And indeed, we found that the number of live-on positive macrophages decrease and correlate in fact with the extent of the plaque. And using depletion model, we show indeed that the depletion of live-on positive macrophages further exacerbate atherosclerosis. And this is particularly because they are important to control the necrosis of the plaque, so the growth of the plaque. So here, really showing that live-on positive macrophages are really important to maintain homeostasis of your arteries, and unfortunately, during hypercholesteremia or aging, their function is altered. So we can maybe try to look at this population could be maybe a biomarker of vascular, a healthy vasculature, and this is what we are looking at right now. I just want to also share that this control of collagen by live-on positive macrophages is not just restricted to aorta. And in fact, we have shown particularly with our collaborator, Florent Genoux, that this is also the case in the lung. So they are critical to prevent fibrosis in the lung. And also we have a very recent story in the skin where we found that indeed in the skin, they also control not just the abundance of collagen, but also the organization of the collagen. And you can appreciate that in absence of live-on positive macrophages, you can see the fibers are disorganized compared to mice which contain live-on positive macrophages. So really showing their importance not just to control the content in matrix, but also their organization. And in this model, we show that this alteration in a matrix have an impact on bacteria pathogenesis, and particularly here is a group A Streptococcus. So just here, another message here is that these live-on positive macrophages we like to propose that they are in fact a master regulator of the tissue matrix through really the crosstalk with different stromal cells. So maybe smooth muscle cells when they are in the blood vessels, but maybe in the skin is through fibroblasts. And this is what we are trying to understand better. So in the field in macrophage, I think now the major challenge that we are facing is to have cells from human cells, human macrophages that mimic these tissue-resident macrophages because they are very difficult, as you can imagine. It's difficult to isolate them from tissues like skin, aorta, for accessibility and to do functional assay. So we decided to develop a method to basically try to generate in vitro these live-1 positive macrophages. And so we decided to look at blood monocytes, which is more accessible, and to find a special cocktail to basically differentiate these cells into live-1 positive macrophages. Unfortunately, I can't reveal the cocktail here, but I'd like to share some of these data where we show that indeed this cocktail X really increased the yield of these live-1 positive macrophages, which express high level of live-1. And you can see now here they have a very beautiful shape and a really morphology of macrophages. And further analysis by transcriptomic flow cytometry really revealed that these in vitro generated cells express hallmark of live-1 positive macrophages, as shown here, CD206, FOLR2, etc. And their transcriptome. So these are the in vitro live-1 positive cells generated. And you can see that they really express all these live-1 macrophage marker. And furthermore, we show that they are functionally similar to this live-1 positive macrophage in vivo. Particularly, they indeed are able to uptake and degrade collagen. And this platform really helped us also to study things that we were not able to do. For example, migration. I'm not sure if I can show the movie, maybe quickly. How long I have? 1 minute. Okay, I will skip. I will show you this next time, or you can come to me and I can show you this beautiful movie. So really, I think here we have the potential with this new platform to really further explore the macrophage biology and their utility for cell therapy. So I would like to finish, of course, to acknowledge my very talented team. So they're all there. I think this is where I become a bit more emotional. Really, they are amazing team, very resilient. I'm very proud of your achievement. It's been a fantastic journey together. So these are the more lymphatic team, the macrophage, human live-1 macrophages here. And then there's some crosstalk between the lymphatic team and the mouse live-1 team, and particularly also I'd like to thank Michael and Gladys, who are amazing to support the infrastructure. I mean, all our experiments, and Michelet. I also like, of course, to thank all our collaborators from Singapore, but overseas, and particularly also all our co-facility in the department and also at the Life Science Institute. And then I have also a very special thank you for two really critical persons. They're always on the background, but they do amazing and facilitate all the research during my journey, Shente and Sirin. I would like to thank you very much for your attention. And I'd like to thank again the department for nurturing, all these years, and to now I'm well equipped to move from Singapore to Nice, since I'm leaving next week, and relocate to France. But I will still be around as NUS visiting prof. Thank you very much.
Thank you so much, Veronique, for sharing your interesting data with us. We have plenty of time for questions, please. Maybe just one quick one. Veronique, have you checked the interaction between pathogens and whether they interact with the live-1 macrophages?
Yeah, very good question. So we know that they seems to be very sensitive to pathogen. Of course, they probably play a role in the host defense, but in the skin, for example, I went a bit fast, but we show that in their absence, because of these changes in the matrix, in fact, in this case, it allow more immune cells to come in because the matrix was more loose. So which allow more immune cells to get into the tissue and then basically to be more protective. So in this case, it was a good think that they were not around because they, because of their remodeling matrix properties. But I think we need to explore this further. And in fact, it's what I will do also in France and looking at these macrophages and their relevance in sepsis in particular. Yeah, Shiva.
Yeah, I think I'll ask you this question in Singapore. Next time, perhaps I go to France to ask you.
Yes, please.
Yeah.
Very lovely.
I mean, the first question may be more serious science. That is, if you culture with a cocktail, the culture the Ly1 positive macrophages, and how stable are they when you remove the cocktails? Do they reverse? And the macrophages are quite kind of plastic. Do they have a kind of permanent fingerprint? They stay there, or they'll
Yeah. So in fact, this was interesting because there was someone in the lab who asked to do this experiment. So we did it, and it seems like once you have added this cocktail early, it's basically will maintain, the phenotype will stays. So I think it's very early, and this is why now we are looking at what is the this cocktail regulating, at the transcription level, and it seems like really it control the transcription of a lot of these markers that are hallmark of tissue resident macrophages. So I think that's why maybe this cocktail is able to really potentiate the differentiation of these macrophages, and it's not required for the maintenance.
Okay. But did you look at the epigenetics of the cells?
Yeah, so this is the next step. Yeah, we can discuss this further. Yeah.
Yeah, that could be the one that actually make it more universal. I mean, that could be a signature.
Exactly.
Epigenetics.
Yeah, exactly.
Well, the other one's common. It's very another question. If all the lymphatics are good, I mean, there's no problem with the macrophages. Everything normal. And what can people do to make the lymph to flow faster?
Yes, this is a very good question, and I'm really for promoting lymphatic as to be well, I mean, for the well-being. So wellness. So it's one, yoga is very good. Exercise. So walking is very important, of course. Running, but even soft, exercise. So yoga, respiration, we, often forget, but when you, inspire and expire, deeply, your lymphatics that are covered, all around the thoracic cage, basically will also be stimulated. And this is very important. Diet is also another, important, aspect. As I show you, if you maybe have too much cholesterol and so forth, might be really impacting, the pumping of your vessels. So I think that's also, and I think I read somewhere also, laughing is also very important because again, you are stimulating your, lymphatic, vessels. So I think we should take care of them indeed, by this little, but I think it's common, to have better lifestyle, right? Like diet, exercise is always, something.
I'm 100% with you with the second question also.
Yeah. Well,
thank you.
Maybe also natural herbs. I think we have an intervention of someone who mentioned about, medicinal, Chinese traditional medicine and so forth. Sure. Thank you.
Thank you so much, Veronique. And we wish you all the best in Nice, in your new position.
All right, we are now ready for the next tea break. So please come back by 3:30. Thank you.
Okay, good afternoon. Thanks for coming back. And today, our next speaker is Professor James McCluskey. So he is no stranger to our department. He has been our visiting panel for our teaching activity. So he has met all our faculty and teaching staff, and she knows about our research. Yeah. Okay. So he has been working on immune recognition, autoimmunity, and antigen presentation. And recently, he has been specializing on a lot of study on mast cells, which play key roles in cancer as well as other disease. So without further ado, let's welcome James.
Thank you very much. I'm like everyone else, really quite honoured to be speaking at this symposium. I've had a very long relationship with the Department of Microbiology and Immunology at NUS, going back for 40 years. Soha Chan was a very dear friend of mine, and in fact, stayed in my home in Perth on a number of occasions, and I used to come and see him whenever I was in Singapore. And then, of course, through the various committees and review teams that I've been involved in, I've met Mike and Nick and Mary and successive leaders. Victor I've known forever. So I feel very close to the department, and I'm really honoured to be here. We're going to talk about this relatively new set of T cells called mate cells, which recognise a molecule called MR1, MHC-related class 1-like molecule. It turns out that there's MR1 restricted immunity over and above mate cell immunity, so we'll talk about both. And I do want to congratulate the department on turning 100. You don't miss a day over 30. I mean, the fantastic place, fantastic achievement. So these mate cells are part of this, growing family of unconventional T cells, which you can think of as sort of being, providers of somewhat more customized immunity. At least the specificity is more customized. If what they do is more of the same in some ways. But here are mate cells here. And a number of these cells have fairly fixed T cell receptors. And mate cells are actually called mucosal associated invariant T cells. The receptor is not invariant, but it's relatively oligoclonal, tends to use the same alpha chain as a J alpha 33 and a restricted set of beta chains. And as I said, it sees a molecule called MR1, which assembles with beta 2 microglobulins, class 1-like, has a cleft. And the ligand was previously unknown, but we now know what the ligand is, and the ligand is an intermediate for microbial ribosynthesis pathway. So what makes this interesting, these cells can be up to 10% of your T cells in the blood. Usually they're closer to 1 or 2%, but even 1 or 2% of the whole T cell compartment is a lot of T cells. And they can be activated by the T cell receptor or independently through cytokines. They don't expand in germ-free mice, so they need bacteria to expand them. And they're activated by bacteria and use nearly all bacteria, but not quite all bacteria. And they provide protective immunity against bacteria and, oddly enough, some viruses through indirect activation. They also promote wound healing, and as I said, this molecule called MR1, although it presents riboflavin synthesis intermediates as the dominant ligand, turns out can also present other antigens. So there's the talk in a nutshell. We discovered that this riboflavin intermediate was what triggered mate cells. In a very odd way, we had 2 strokes of luck. The first was that you didn't need whole bacteria to stimulate the mate cells. You could do it with culture supernatant. The biochemists in the audience will say, well, there's 2 orders of magnitude luck straight away. Kind of fishing a supernatant is a lot easier than fishing in a cell lyso. Secondly, when we went fishing with recombinant empty MR1, we got a pretty clean peak on the mass spectrometer. And it was slightly yellow. Not quite as yellow as that. This is straight from Google. But it was slightly yellow, and we thought, well, what could be yellow? And of course, vitamins are yellow. And we thought, is it possible? And we made a series of mutants of the riboflavin pathway, and some of the enzymes, when they were knocked out the capacity of those bacteria to stimulate mate cells, pointing the finger at the ribosome pathway as the dominant ligand for mate cells. And if you look at the pathway, it turns out it's not as simple as it sounds. It turns out that the enzymatic mutants all point to this compound 5-aminouracil as being essential, but it's not enough. It doesn't stimulate mate cells by itself. It needs to combine non-enzymatically with small intermediate metabolites, principally methyl glyoxyl or glyoxyl. So nothing to do with the riboflavin synthesis pathway. So this does, rather, but not this. And they go on to produce these losers, of which ultimately riboflavin is derived. But along the way, there are somewhat unstable pyrimidine adducts. The one end is this thing called 5-OTRU, or is a version of it called 5-OERU, where the two of which are hydrogens. So it turns out that MR1 is capturing an unstable intermediate in microbial riboflavin synthesis and presenting this to T cells, to Woodard-Jeffs. And that presentation, we've now solved the structure. And here's the 5-OTRU in green, and the grey is the MR1. And it turns out it forms a shift base here. So this is the 5-OTRU. There's a lysine on the internal helix of MR1 forming a shift base. There's the aldehyde in 5-OTRU causes the shift base or guides the shift base formation. And then there's a funny little tail to the molecule here, the ribotyl tail shown here in the structure, which picks up in the air a little bit and is available for T cell recognition. And the T cells that recognize this, remember, they're semi-invariant, and so all the structures we've made, because they're all overlap, show exactly the same mode of recognition. Here's a tyrosine, position 95 in the CDR3 alpha, the alpha chain of the T cell receptor. The CDR3 alpha comes from the J region of TCR alpha gene. The only J regions that are used are J alpha 33, 12, and 20. They all have a tyrosine at this position, and they are the only ones with a tyrosine at that position. So this is hyperselection of a single TCR with a common tyrosine at position 95, stereotypic recognition of this ribotyl tail. So it's almost an innate-like receptor. And then when you go to the list of bacteria that do stimulate mate cells, they all have a riboflavin pathway when you go to the KEGG database. And when you go to the ones that don't stimulate mate cells, they don't have a riboflavin pathway. They scavenge it from other organisms. So the message is that the immune system, which specializes in seeing things that are foreign, that are signatures of microbial invasion, that are non-self, is pretty promiscuous, pretty ubiquitous about what it'll do here. And here it's determined that, okay, well, we don't make vitamins. We're mammals. We don't make vitamins. We get them from our Wheaties and our breakfast cereal in the morning. But bacteria and yeast do make vitamins. And therefore, the vitamin synthesis pathways are targets of immune recognition. We were able then to make tetramers of MR1 loaded with the antigen 5-OTRU. We sent them then to the Veronique tetramer facility, which when I last looked is still being funded. And when you stain peripheral blood T cells with these tetramers, you get a whole bunch of mate cells. I'm not sure why there's so many in this particular case. They're generally CD8 positive in humans. There are some double negatives, a small number, and even a small number of CD4 positive mate cells. Mostly CD8 positive, remember CD8 is a co-receptor for class 1. MR1 is a class 1 like molecule. Mate cells are abundant in humans, but a bit rare in the mice that we've worked with, the inbred strains of mice that sit in very clean cages that are very meticulously cleaned every other day. They decline over age, with that, there's a decline in mate cell activity over age, and they're found in nearly all tissues. And it's probably a slight misnomer to call them mucosal associated, because they're found in the lung, in the liver, in uterus, and of course they are found in the gut. But they're kind of barrier cells, and there's not a lot of them in secondary lymphoid tissue. They seem to mostly sit in tissues, and they're innate-like, they're easily activated. Many of them have a memory phenotype. They do protect against infection. And so here's just a model infection where we infected mice with salmonella, that infected them in the lung, not a natural site of infection, but pretty good for experimental purposes. And just look at the way the mate cells expand. There's 36% of all T cells in the lung following infection, and this is what happens in an MR1 deficient mouse. You get no amplification of mate cells, utterly MR1 dependent expansion. And then if you do Legionella, and this is an actual site of infection for Legionella, same thing, massive expansion of mate cells. And then if you do systemic infection with Francisella florensis, Borrelia pathogen, you can see again expansion in all the organs. This is a systemic with infected. But for 12 and 37% of the only T cell innate cells following Legionella challenge. And they're long lived. You go out to 300 days and there's still a lot more mate cells than there were at the beginning, pre-infection. So they stick around for a long time. They have a kind of tissue resident memory aspect to them. And they do protect, but the protection is not what you'd call dramatic. It's kind of an author's nightmare, and, Christopher Mill for reviewers to be able to say, well, that's not very big, is it? So here's the protection. These are your MR1 deficient mice. This is colony forming units. It's Legionella. It's in the lung. And then here are your MR1 positive mice. It's about a half a log protection at CFU. In Francisella, again, it's between a log and half a log in the liver, in the lung, in the spleen. And that protection translates into survival in the case of Salmonella, where this is wild type animals with mate cells, and here are your MR1 deficient cells. So they expand, they protect. And that protection is dependent on interferon gamma. So in this experiment, we've taken profoundly immunodeficient mice, grade 3 gamma C knockouts, that, as you know, adaptive immune system, transferred mate cells into the mice after a bacterial challenge, and asked, well, in the absence of any other immune system, are mate cells healthy? And they are indeed. There's the survival of the animals with mate cells. There's the survival in purple with mate cells but an anti-MR1 antibody. And then there's the untreated. And if the adoptively transferred T cells, which are mate cells, purified mate cells, are from a gamma interferon knockout mouse, you get no protection. Whereas if they're from an IL-17, they're perforin, they're a TNF, or neochildren, or granzyme knockout, they're basically indistinguishable from wild type. So that's protection. I'm not going to show you the data, but it turns out that in influenza infection. You can also see protection. It's again half a log. Influenza don't make riboflavin. The pathway we consider to be indirect activation of MATE cells that are sitting in the lung. The lung is an organ rich in MATE cells. The other organ that's very rich in MATE cells is the liver. We also did a transcriptomics analysis, as did others, and showed that many MATE cells have the transcriptional profile of the wound healing and tissue repair, shown here with these genes here, as well as their pro-inflammatory cytokines, shown here. Others have gone on to show they promote wound healing, and in fact, Ken Hinks, who was my postdoc, has got just published that in a sterile model of lung damage using bleomycin, nasty cytotoxic drug. If you've got MATE cells, you do much better in terms of healing of the lung following the toxic insult of bleomycin than if you don't have MATE cells. And there are other models from other groups. So they have a homeostatic role as well as protecting us from microbes. Now, ML1 is relatively monomorphic. But when you dig deeper and do a population study, there are some natural polymorphisms. And at least 6 allelic variants have been described. Most of them have mutations outside of the cleft, shown here. And they make very little difference to the function of that particular allotypic form of MR1. But these two alleles here, MR10401 and MR10402, they both have a mutation arginine to histidine in the position 9. So that's here, and has the potential to impair antigen presentation. Turns out in Melbourne we found a patient who's homozygous to this, and they have an odd phenotype of vasculitis, genital warts, and candida. When you look at the functional behavior of these different alleles, just here their expression following transfection, which is matched for a marker gene, you see the two MR104 allotypes, which have the R9H, both express much better than the other allotypes. You can see that on the fact plot here as well. But they don't present 5-OT-RU to MATE cell. So here you can see TNF production, interferon gamma production in response to APC pulse with 5-OT-RUs, and here you can see no signal at all from the MR104 alleles. And what appears, this is just the same data presented now as MFI, slowly in the percentage of cells. So they're not presenting the canonical antigen, but they're expressed perfectly normal, or in fact better than normal. And so we wondered if they presented antigen of some sort to a group of non-MATE cells called MR1 T cells. So these are T cells known to be MR1 restricted, but for which there is no known antigen. And to our great surprise, this clone called MC7G5, described by the Carter fit, described as a pan-cancer T cell because it killed nearly all cancer of APC that were tested by Andy Searle and his team. This clone, for which the ligand was not known, and it was the holy grail if it's a truly pan-cancer T cell, it recognizes R9H very, very well. And it recognizes wild type MR1 moderately well. It doesn't see at the MR1 with a mutation at the site of the shift base or null alleles. This other clone, which we produced in our lab, has a very similar phenotype, and which you are getting some presentation endogenously. And mate cell, a canonical mate cell, for way of study, is also able to recognize this antigen. So this is endogenous. These are just APC. They're not pulsed with antigen. So whatever's being presented is endogenous. And so we did the mass spec, the purification, the biochemistry of the mass spec. I'm not going to show you all that because it'll murder you at this time of the day. But it turned out the antigen is pyridoxal. Pyridoxal is vitamin B6. So here is an analysis of the capacity of various forms that are known as vitamins, but pyridoxal, to upregulate MR1 on antigen-presenting cells that have got low levels of MR1 because there's no antigen there. And what you can see is how good the blue one is, 7.6FP, which is our control upregulatory molecule. It's a derivative of folate. We know it binds these molecules very well, upregulates MR1 better than any of know. But we're seeing upregulation with pyridoxal. And we see even more upregulation with the R9H molecule. So this mutation that couldn't present the canonical antigen seems to be presenting or capable of binding pyridoxal very well. And we've now shown unequivocally that that particular mutant or allotype of MR1, R9H or MR104, is naturally presenting, constitutively naturally presenting pyridoxal. Now, interestingly, they don't present pyridoxine or pyridoxamine very well. There's some weak presentations of the phosphorylated version of the oil. So they're very fussy about, they want this one, these ones aren't good enough, and the control works very good. You make tetramers of R9H or tetramers of wild type with pyridoxal and go into normal blood donors. You can see very small numbers of these cells in peripheral blood, and they're easily enriched using the tetramer and expansion technology. So they are natural T cells. So this is intriguing, and that I don't have a physiological endpoint for this. The principle, however, is that MR1 can present other antigens, and where and when does that matter? So you need to find out. The possibility that the so-called pan-cancer T cell that Andy Sewell described, that recognizes pyridoxal, really is a pan-cancer T cell, can't be totally discounted, because we now know that cancer cells suck up huge amounts of pyridoxine from meat. They're very hungry, and they're churning out a lot of pyridoxine. So it could be simply a matter of there being an antigen preferentially present in high concentrations of cancer cells, not found in wild-type cells. So it's now emerged that MR1 binds a very diverse array of small molecules, including those derived from microbes that I've described earlier, those derived from the host, drugs, and diet. And they include nuclear-based adducts, I'll talk about in a moment, various drugs we've published on previously. There are compounds in cigarette smoke that it seems to be able to bind. I've just shown you as a control this form of ferromesetic 6FT folate because I mean, vitamin B6, the data I just showed, in Japan, a group from IFREC had shown that sulfated bile acids can be presented by MR1 to MR1 restricted T cells, not innate cells. And of those, we become interested in the possibility that highly reactive compounds other than methyl dioxide might be able to complex with 5-ARU. And one of the ones you've looked at is malondialdehyde, because it's very reactive. It's found as a result of lipid peroxidation with prominent products and prostaglandins reduction, the biomarker therefore of oxidative stress, reactors DNA and protein. So in the quest to identify other ligands of potential physiological significance, we've taken a chemical approach to say, well, at first principle state, this is a possibility, because this is abundant, physiological, so is this, and so is that. So this come together with that, that's all. And it turns out it does. You can get a compound called N-ribitol 9-formyl 7-deazoxanthe, which we call RFCX for short, which binds to MR1. There's binding there. It's solved the structure, and it turns out you can generate T cells to this fairly easily. They're present in peripheral blood of normal donors. These two T cells are fascinating. So this is a titration of activation of these two T cell clones, pulsed with RFDX from MR1 APC. They both have a tyrosine at position 94, not position 95. And when you take clones that do have a tyrosine at 95, they're not recognizing the antigen. So we're seeing differential recognition of these compounds created in this odd synthetic way to create MR1 ligands. You'll notice that this compound has a ribitol tail, very like the ribitol tail on 5-APRU. And indeed, all of the clones that I just showed you recognize 5-APRU. And the reason why the tyrosine 94 also seems to be able to see RFDX is the change in this position in the tyrosine, which sits like this if you're a tyrosine 95 receptor, that it scribbles around, gets closer to the antigen if you're a tyrosine 94 receptor. So there's a structural basis for this differential antigen recognition. Now, others have also taken a first principles chemistry approach to identifying new ligands. And Gennaro de Lavilla's group, many of you will know Gennaro, have looked at stress-related compounds created particularly from molecules that have certain credentials that predict that they should react with malondialdehyde. So he came to the same conclusion, and he discovered that this antigen here, which we call M3A, stimulates T cells. And this compound is created by adenine complexing with 3 malondialdehydes to create this compound here, M3A, M3-adenine. And this compound will rather efficiently upregulate MR1. These are parental cells. These are the null cells here. And these are null cells transfected with different forms of MR1 as negative control. And so You can upregulate MR1, which implies that M3A is binding efficiently to MR1. And you can make tetramers of this MR1 M3A antigen, and you can find these T cells ex vivo in peripheral blood. You can enrich them. When you enrich them, they're all CD8 positive, and they are activated by M3A in this dose titration here. They're not activated by acetylcitrate or by 5-FTAU. These cells are conventional CD8 positive T cells. They're not mate cells. They're recognizing MR1 and this compound called M3A, just like the previous cells were recognizing RFDX. So this, I think, is telling us that there could be a deeper story around the immune regulation that MR1 conducts. The big story is that it's the antigen presentation molecule for riboflavin synthetic intermediates that activate mate cells, which have a very stereotypic receptor and quite stereotypic phenotypic functional characteristics. The other story is that MR1 is quite a big pocket, and these are small molecules. And this pocket can bind other molecules that are structurally related but not identical, that are formed from highly reactive metabolites, some of which condense with 5-ARU, which is from the riboflavin pathway, some of which condense with other molecules that are natural in living cells, like adenine. Story that I think is yet to unfold. So, I've kept this fairly short, but I just want to summarize what I think is going on with mate cells. It's been an interesting question to me as to why these are preserved through most mammals. Not all mammals, but most mammals. Mouse mate cells will work on human MR1. Human mate cells will work on mouse MR1. Highly conserved. The MR1 molecules across species are more like each other than MHC molecules are across species. And they have to be more like each other because they're binding the same antigen, 5-FTAU from the riboflavin pathway. They seem to play cameo roles, these mate cells. The antibacterial immunity we saw is half a log to a log in controlling CFU. I think it's interesting, however, to contemplate that when you remove other layers of the immune system, as we did with the RAG gamma knockouts, and just leave it up to the mates, they step up to the plate and they can control the infection. Is this the sort of classic redundancy that Professor Taniguchi talked about earlier today, where you have redundancy that can be absorbed by the rest of the immune system, but if the rest of the immune system is not working so well, you're immune depressed, you're immunodeficient, you're elderly, you're very young, you've got chronic illness, then mate cells can make a difference. I think of it as a kind of cliff edge immunity. If you're just on the edge of the cliff and it doesn't take much to push you over so your legs are pointing upwards instead of downwards, as Zinke Nagel likes to say, then mate cells could make a difference. And they're selected through evolution for that purpose. But perhaps the other reason they're selected is they're polyfunctional. We know they make a broad range of cytokines. They accelerate wound healing and tissue repair. There's some evidence that they're important in barrier integrity, that surfaces are leakier if you don't have mate cells. And then lastly, MR1 has been hijacked in an odd kind of a way to find other antigens apart from riboflavin synthesis intermediates, antigens that are small ring compounds with a tail of some sort that's attracted to T cells, and the T cells that are seeing these antigens are mostly conventional CD8 positive T cells drawn entirely from the adaptive repertoire. When could that be important? Well, one possibility is cancer, and I alluded to Andy Sewell's putative pan-cancer T cell. Another possibility is in maintenance of homeostasis. Over the last 15 years I've worked on this project, had help from an awful lot of people. I do want to recognize my single biggest collaborator, Jamie Rosjohn from Monash, who's done all the structural work. And Lars P. Nielsen, who made the discovery of the riboflavin synthesis source for mate cell antigens. Thank you.
The floor is open for questions.
Jim, wonderful talk, and thank you so much for joining for this event. In diseases that are associated with intestinal dysbiosis, things like Crohn's and so on, what's happening to the mixed cell biology in that context? Because, of course, the bacterial communities are changing significantly. And
it's a great question. It's an intuitive question, isn't it? I wish I could say they're the cause of ulcerative colitis or Crohn's disease, but the literature is patchy. There are some changes in mate cells, the phenotype of the mate cells in those lesions, but there's no strong evidence to suggest that they're driving the inflammation you see in inflammatory bowel disease. I don't think that the book is totally finished on this, because there's such a pile-on in those inflammatory bowel disease. If you could get to patients early in the development of the disease, and you could get the right sort of biopsies where you've got paired normal and so it's aching to be done. It hasn't been done properly.
So, good. Thank you very much for the talk. Very interesting, intriguing subject. And I personally don't know much about myself, but I was interested by you finding out the anti-cancer efficacy of these cells and the pan-cancer efficacy. And what would you guess are the strongest hurdles to utilize those cells for immunotherapy, for cancer immunotherapy? And are they actually sensitive to the usual cancer immune evasion mechanisms cancer actually displays?
Yeah, so there are several spin-out companies now whose portfolio is harnessing MR1 restricted T cells for potential anti-cancer therapy. I think the biggest hurdle is going to be specificity. If the antigens that are being seen are also present in normal cells, you need to find a therapeutic window where you just knock off the cancer cells and not the normal cells. The pyridoxal presentation we saw, you can find conditions where only cancer cells are knocked off. So, we're growing those cells in culture medium. It's very rich in vitamins. They're taking up a lot of the vitamins preferentially compared with normal cells, rendering themselves visible to pyridoxal specific T cells. Is that going to happen in vivo? And I think that's the great, challenge for the spin-out companies is specificity. Immune escape, well, yes, I think the answer is going to always be yes. But one thing that's interesting is you probably don't have the problem of getting these T cells into tissues that you might have with conventional CD8 killers, because their chemokine homing receptors say, I'm off to the tissues. And I showed you the distribution earlier, and you don't find many of these in themselves. They like to be in tissues. They're frontline, sentinels. So I think that's a good advantage actually, that they'll naturally go into organs, particularly the lung and the liver, which are two big places, you want to deal with the liver cancer and lung cancer if you can.
Yes, very nice, James. Just two questions. One, I was intrigued by the fact that you showed that the mate cells decline with age. I was wondering whether is it because like the production is decreasing, with age, or is it maybe maintenance, and it can be related to microbial changes, over age, like dysbiosis. And the second question is whether ML1 can bind MDA LDL. Like MDA, the low density lipoprotein has been shown to be modified, and for example, you can find MDA LDL. So I was just wondering whether it can bind to ML1.
So what is the mechanism by which the cells decline with age? I don't know the answer. I mean, it's just an observation. And whether it's related to change in microbiome, stiffening lymphatics, or the sort of general outcome of the depression in the immune system that takes place with age, I don't know. As for what the binding capacity of MR1 and low density lipoproteins, I doubt it very much. The cleft is not like the classical class 1 cleft. It's got a sort of small A pocket and a smaller shape pocket, and it's tap independent. So it's not using the same chaperones to antigen load that class 1 uses. And the other thing I can tell you is that when I said we were lucky that we found this antigen, it's because everybody else was expecting a peptide, and they had all of their size exclusion chromatography and everything geared to 1,000 Dalton peptide. And these molecules are 300 Daltons. So they were reasonable. So that's just an intuitive answer, Veronique. I can't give you a definitive answer.
Thank you. Thank you.
Okay, last but not least, we have Shee Lok, our next speaker. She's at the Emerging Infections Program in Duke-NUS. She did her MSc with our department under Prof Malik. After that, she did her PhD in structural biology, and then postdoc overseas, continuing on structural biology. So she has made a lot of new discovery regarding dengue virus, morphology of viral particle, neutralizing antibody mechanism. So we'll pass it over to her.
So, thank you for inviting me. It's nice to see a lot of familiar faces. So I did my PhD, my master's here 29 years ago. I feel ancient. Yeah. But it is important for my career because all of you know I'm a structural biologist, but actually I do know immunology and virology, and that's because of the education in Department of Micro. And that helped shape my career in the sense that I can interpret structural in a biological sense. So it is very important that I can write paper well and I can write grants really well too. Okay, so after studying here, I went to do a PhD in crystallography, and after crystallography, I did a postdoc with Michael Rossman, and I learned crystallography and cryo-EM, single particle analysis, and I came back here. And I also went to Watcher's lab to learn cryo-EM. So the evolution of the cryo-EM, we start from cryo-EM single particle analysis and started off in 10-ish resolution, and then now is 2.8 angstrom or 2.5 angstrom or even higher resolution. So, and then next, those are very symmetric particles, but a lot of particles are not symmetric. It's geomorphic. So then you have to use cryotomography, which is much harder. So we are doing that now. And then we are also setting up equipment to start solving structures inside the cells. So you can see exactly what are they doing inside and what are they complexing and things like that. It's very exciting development. But right now it's still, what I'm showing you is still in vitro purified virus and microsome, and, but analyzing the fusion of the virus, when they are in the endosome. Okay, so it's a tomography studies of dengue fusion process, so you should not expect high resolution structures. So what is dengue virus? It is an important pathogen in Singapore. It affects about 400 million people per year worldwide. It's a mosquito-borne disease, positive sense RNA virus. There are 4 serotypes ranging 1 to 4, and the disease range from mild dengue fever to the more severe dengue hemorrhagic fever. Okay, so this is the dengue virus infection cycle. Mature virus bind to a series of receptors. It's still kind of unknown which are the receptors, but it could be more than one. So it binds to a receptor and then it gets endocytosed. And then in the endosome, the lowering of pH causes the virus to undergo major conformational changes from dimer to trimers, and that induces fusion between the virus and the endosomal membrane, releasing the RNA. And then the protein translation starts, and then you have viral replication, and the new virus particles are assembled in the ER. So this is immature virus. It has additional protein on top, PR, protein binding to the E, and that is to prevent the virus from fusing back in, because it has to go through an acidic compartment just like the endosome. So, and it goes through the trans-Golgi network. The lowering of pH causes the virus to rearrange into dimers, actually just like this, except it has an additional protein bind on top of the fusion loop. And then in this pathway, the furin then cleave off the PR from the virus surface, but the PR stays capped on top. And only when it is released to the outside, when the pH increases, the affinity between the PR and the E protein drops, then the PR fall off the particle, making the mature virus, and then it starts the next cycle. So I'm interested in this stage. So very little is known. Okay, so the major surface protein of dengue is the E protein. So it is important for entry like receptor binding and fusion to the endosomal membrane. So this is a crystal structure solved by both Steve Harrison's lab and Michael Rosman's lab. So it is dimers. So this is one protomer. It has 3 domains, domain 1, domain 2, and domain 3. And this is the dengue virus structures on the virus surface. There are 180 copies of them. So they are arranged as dimers. So 3 dimers lie parallel to each other, forming a raft. And there's 30 of such rafts arranged in a herringbone pattern. Okay, so these are the important parts of the E protein that are important for fusion. So the tip of domain 2 is interacting directly with the endosomal membrane. And this hinge is thought to be important to flex the whole of domain 2 so that the fusion loop can reach the endosomal membrane. And in the post-fusion structure, domain 3 actually moves over here by 70 degrees. So it must be important. So this is the E protein post-fusion trimeric crystal structure. It appears as trimers, and all the fusion loop are assembled at one end. Okay, if you remember this, so this is a dimer. In order to change it to a trimer, there should be massive structural changes. And nobody knows how it works. Okay, so this is suggested entry mechanism of mature dengue virus into cells. So it's from the paper published by Steve Harrison's lab. So this is the E protein dimer on the virus surface. It binds to the receptor on the host cells. And then when it gets into the low pH environment in the endosome, the E protein flips up, showing its fusion loop. And the fusion loop then interacts with the endosomal membrane, and the E protein start trimerizing. So in this stage, the domain 3 start twisting, bringing the 2 membranes closer together, and then it fuse and open up a pore, and then the viral RNA can be released into the cell cytoplasm. But in actual fact, this whole entire mechanism you only know about the structure here and here because they have been solved by crystal structure. We don't know anything in the transitional state. Okay, so we wanted to understand how this works. So we're going to do cryotomography, but our experimental work are all done at 4 degrees Celsius, where the two lipid, the viral membrane lipid and the endosome, the liposomal membrane are all done at 4 degrees Celsius, so there is no hemifusion. That means we probably can see only up to this stage before the lipids start mixing. Okay, so the first thing we did is we characterized the virus interaction with different types of liposome at different pHs. So we make liposomes that mimic the endosomal membrane and the plasma membrane, at least in terms of charges. So in endosomal membrane are negatively charged, plasma membrane is neutral charges. So in order to make them negatively charged for endosomal mimicking liposome, we add PS here and BMG here. So these are the different pH. It gradually decreased, mimicking from the early endosome to the late endosome. So this is an amplification of this part. So I'm going, I don't know about this. I can't see it well. So I'm zooming in this. Okay, at pH 8, liposome are the ones that are empty. And then the virus is the one that is regular size and we have the dark center. So at pH 8, all viruses are not interested in the liposome. And then when you go down to pH 6.5, we see that the virus are attracted towards the liposome, but we don't see any physical interaction. So at to the neutral charge membrane, the virus is still not interested in the liposome. At pH 6.5, we do see some connections here. I think the E protein has flipped up and interact, but it only interact at the phase where it sees the liposome. So actually the liposome does do something to the virus, so it attracts a fusion loop. Whereas the other side where it doesn't see the liposome, nothing happens there. And then at pH, so for neutral charges, there is a little interaction, but it's rarely observed. So in the late endosomal pH 5.5, the virus surface ruffled up. I think all the E protein are loose. And then you can see a lot of interaction with liposome. They actually just aggregate. And for the neutral pH, the virus prefer to interact with virus and not to the liposome. So we think one of the mechanism of why it has to go through this gradual lowering of pH is it needs, in order to direct fusion between the virus and the endosomal membranes, you need to go through this because at this pH, the virus get attracted to the endosomal surface, but not yet interact. And then when it goes down, then it starts interacting. So if you go straight down from pH 8 to 5.5, the virus will be aggregating with each other and not interacting with the endosome. So for cryo-EM structural studies, we think this sample would be easier to solve because most of the surface is still smooth except the part that is looking at the liposome. So we are formally cryo-EM single particle analysis people. So we thought this is easier to solve. Perhaps every interacting interface is exactly the same as each other, so we can average them. But later on, I will show you that's not the case. So we can only solve it by cryotomography. Okay, so this is the sample that we used for further work. Okay, so this project, I was very ambitious. This was done like 10 years ago where the technology is still not quite mature yet in terms of the hardware. Okay, so this protein is really small. It's 50 kilodalton. And during that time, the detector was a Falcon 3 detector and it's not sensitive. So we have to put in something called a Volta-phase plate to increase the contrast. But it is really hard to collect a Volta-phase plate data set. Okay. And you cannot use it to solve to high resolution because there's something called a change of phase with every different micrographs, and it's very hard to figure out the phase change. So, but nevertheless, we can see the density. And what we did is after we collect tomography, is like your CT scan, we make a tomogram, which is a 3D tomogram. After you get the tomogram, you can see through the zinc cycle. I it's just like children doing tracing. If you see a density, you draw a circle, and then you keep following in every Z slice. So we drew the density. And then what we did is, we saw three types of density. And then we fitted in the structure of the E protein just to compare that dimension. It's not to interpret the structure. So we saw three types of density. The first type, the dimension fits that of the E protein. You can see the E protein fits very well. And the next one, the width is correct, but then the length is much longer. And so this is a structure of the E protein on the virus surface. You can see there's a stem region. So we think what is happening is the stem region is loosened. So it is released. And so you can see it from here, stretch out and release. And so you can see the E protein moving up there. And then this one is much thicker. So when we fit in the trimer, the width is correct, but it is still a lot longer. So we think that the trimer also has its stem region extended. Okay, so that is one E protein density. So how does it look like in the interacting interface? So we show you different tomograms, and that's because the liposome size is different, and they interact in a different manner. So, pink cylinders are the thick density, that is the extended trimers. The green one is the extended E monomers, and then the blue one is the non-extended E monomers. So this is the side view of the interaction, and this is looking down the virus. So you can see none of them looks the same as each other. So that's why cryo-EM single particle analysis, where you average all interface, does not work. It's not averageable. So, but from the segmentation, you can see something that is common. The extended monomer is lying at the peripheral of the interacting interface. And so is the extended trimers. So I guess it makes sense. So as you move out from the center outwards, the distance between the liposomes and the viral membrane increases. So in order for the E protein to reach it, the stem region will have to extend. Okay. And how did the trimer form? So we think it comes from the extended monomers because it is in the vicinity of where the extended monomers are. Okay, so, well, like I said earlier, this is a photophase plate image where we cannot do subtomogram averaging to solve it to higher resolution. So we want to do that, and we collected the data. So that involves DBS, Duke-NUS, putting in money to buy a $2 million detector, so we can image that. So this is a tomogram with at the time, new K2 detector. You can see virus interacting with liposome. Okay, so this is the zoom in. So this is the virus. This is the interacting interface. This is another one. You can see the connection of the three proteins. Okay, so this is a Z slice of a tomogram. You can see the extended E monomer, the non-extended E monomer, and the fatter trimer. And these are just a section of other tomograms. Okay, and then we collaborate with Nisha to do unsupervised classification and subtomogram averaging to get different classes of the structure. So my postdoc is very hardworking. So he bought like 6,000 subtomogram of E proteins. So from the liposome downwards, because the stem region is very flexible, we can only average something that is legit. So we boxed here and that and that. And then we classify them into three structures. So we saw three. One is E protein density that is perpendicular to the liposomal membrane. And then next is one that is tilted. And we found one that is very interesting. It's a trimer. That's the fat density. So when you see from the top view, the three form an exact trimer. We did also impose C3 chemistry so that we can get it to 14 angstrom. Okay, and the first thing we are interested in is to find out what what is in this primers? Is it E protein, pre-fusion E protein, or post-fusion E protein? So we treated to compare the length. So you can see that the pre-fusion fits better than the post-fusion. This is much shorter. And then when you look at the density, you can see that the domain 1 has very poor density. The rate charging. So what we think could be happening is domain 1 could be very important in fusion. So Yoga Mundis in his recombinant E protein crystal structure, this is the pre-fusion and that's the post-fusion. It is crystallized in the absence of both the viral membrane and the liposomal membrane, same membranes. You can see that domain 3 moves over here by about 70 degrees, but our tomogram, it comes from the virus where you have lipid and also the liposomal lipid. So it is possible that domain 3 has unfolded so that it can help the domain 3 to move over here. So domain 1 is not just a domain that links the domain 3 and domain 2 together. It has actually maybe a function. Okay, and then we compare our primer. This is our primer to the post-fusion primer, just different leaves. You can see from the top view, the 3 protomers are far apart from each other. So we call our structure an open primer confirmation. Okay, so the next question is the open primer structure reversible by pH? So what we did is we conduct this assay, virus liposomal fusion assay, where the liposome has a fluorescence dye, DID dye. So when it fused with the virus, due to the dilation of the DID, it will release fluorescence. So what we did is we just put the virus and liposome, we mix them at different pH. So and we have some conditions that we did back neutralization back to pH 8. And after we finish mixing, we'll either keep it at 4 degrees Celsius or increase to 28 or 37 degrees Celsius to observe fusion. And we can see when we back neutralize, we don't see any fusion here. So maybe some structures has been reversed. So, and then we conducted, again, cryotomography and segmentation. And we saw that, this is the low pH structure where you can see primers here. And then when we back neutralize, we do not observe any primers. Is it real? So we know that the trimers, the open trimers are reversible when the pH is raised back to 8. And when it did that, it is unable to fuse. So we know that our open trimers are essential for fusion. Okay, so this is our updated fusion process on stages prior to hemifusion. So this is the virus. When you lower to pH 6.5, we do see the virus being attracted to the liposome. What we think is happening is at this pH, the histidines are protonated, so it becomes positively charged. So it will move closer to the liposome. And pH 6.5, some of the E proteins flip up and interact with the liposome. And some of them actually has the stem region extended. And these extended protomers then interact to form the open primers. And domain 1 is starting to unfold so that it can then twist back the 2 membranes. So this is the hypothesis that was suggested previously by Yoga. Okay, so in summary, at low pH, dengue virus has affinity towards anionic membrane, so that fusion is directed between virus to aerosomal membrane instead of virus to virus membrane. So at low pH, some E proteins have released their helical stem regions to form extended structure to interact with liposomal membrane. And the extended trimers could be a result of extended monomers interacting with each other. So we have the low resolution E protein open trimer fusion intermediate structure. And we think domain 1 is important because it may facilitate the movement between domain 2 and domain 3. And we show a proposed fusion, updated proposed fusion process at stages prior to hemifusion. And so this involves a lot of people because it has been done over 10-ish years. So Justin did a lot of the initial work, sample optimization. Aaron did the segmentation and subtomograph leveraging. And Misha's lab helped us collaborate with us with subtomograph leveraging. And now I'm working with machine learning people, system. So we are going to do automated. The limitation is to manually box all this individual density in the colloidal. So Tristan's lab, together with Tristan's lab, we are going to train the machine to do machine learning to automatically pick those density so that we can have more particles and increase the vibration. And Tristan and Mark's lab are also in there. So these are the grants that's sponsored this research over the years. And and the last thing is I want to advertise that in October we are having this HCD Flavory Conference in Duke-NUS. If anybody is interested, we can register for it. Okay. Any questions?
Thank you, Shee, for a really interesting talk. So one of the things I've often wondered about is what happens in the context of the mosquito, because the midgut of the mosquito, which is supposed to be the site of dengue infection, tends to have a very basic pH. You can go up to pH 9. So how does that fit with some of the modeling that's been done in terms of how fusion is taking place?
Well, that's very interesting. I don't know. Actually, I mean, that would be an interesting thing to look at. I mean, we're going to set up the in situ structure, infrastructure. But however, we are not so ambitious yet. We are looking at single cell, not the whole organism. Yeah, but I mean, eventually, probably we'll get there when the technology in place.
Thank you.
Any other questions? No, it's not pH 7. Depending on which compartment you are, the pH changes. Okay. The first slide, right? Yeah. I did label the pH. Here is, well, it decreased from 6.5 to 6. Here is pH 7. And then here is pH 6.7 up to 6.0. Depending on the compartment, it changes. I think cytosol is probably 7, 7. Yeah, yeah. Yeah, I think the interaction of virus with receptor is the extra, well, on the surface, so it'll be 7.4 something. It's neutral pH. Neutral pH. Yeah. It can attach to receptor. And then when it gets endocytosed, the ATPase will start pumping down the pH to 6 point something. Oh no, it's the cell. Cell endosomes are that pH. Yeah, it helps the cells to digest whatever they have taken in. Any more questions?
So Shee, maybe a technical question that you can educate us about CLIA-EM. So besides looking at viral structure in the endosome, does that mean future development we can look at viral protein at different parts like the nucleus?
Yes, you can do that. So I am a little bit not that ambitious. I want to solve things that we can actually see. So right now, the technology is you can cut the cells. So the problem with cryo-imaging of cells is it's very thick. The electron cannot penetrate. Very thick. And then you need to nail it down to 200 nanometer in order to see, in order to be imaged by EM. So we bought this thing that we can nail it down. So, for right now, all the high resolution structures is that of tubulin, flagella, ribosomes, those that is gigantic, that you cannot miss, and the reviewer cannot say you're solving rubbish. So, but if you want something smaller, you have to label it with fluorescence or something. So when we nail, we can track where the fluorescence is. So we nail only we keep only the lamella that has the fluorescence. But the fluorescence milling machine is a confocal machine with a light microscopy. So when you move it to a cryo-EM, which is like, the magnification is much higher. The precision of the position can be lost. So because the confocal is like a light microscope, 100 times at the most. And then when you go over the other side, it's 100,000 times maybe. So the precision is gone. But then now people are developing labels and things like that to dual label it, like it has fluorescence and gold particles. So then you can correlate the two. Yeah.
Okay, if there's no more question, let's thank Shee.
All right, so that concludes the last talk for this symposium. So we are just on time, very good. But we still have one highly anticipated segment. But before that, let's join me together and thank all the speakers for this exciting talks throughout the whole day. And I hope all the participants have a chance to speak to the speakers and discuss more during all the different breaks and lunchtime, right? So now I'll move on to the almost last segment, which is the Posters Prize presentation. And I hope the winners are here, right? So let me welcome our HOD, Professor Kevin Tan, to give out the prizes. So while he's coming up, I'd like to thank the judges who help us with judging all the posters. Those are great posters, so I think they must have a hard time trying to choose the 4 winners, right? So join me to thank our Associate Professor Taniguchi, Associate Professor Chris Sham, Dr. Lo Jin Xiong, and Dr. Chen Kai-wen. All right, I have to read. If I mispronounce your name, you can try to correct me later on, but please come on stage. All right, so all the 4 winners, they are equally good. That's what I was told, so they should be, right? So let's start with the first one. Poster number 9, Donald Heng Rong Ting from Prof Sylvia Alonso Lab. Are you here? Okay. All right, so the second winner, poster number 17, Ao Zhen Qing from Professor Justin Chu's lab. All right, the third winner, poster number 20, Chua Wan Zhen from Prof Chris Sham lab. All right, so the last winner, poster number 30, Tong Jie Xing from Prof Tan's lab. right, so Prof Tan, please stay on the stage for a bit. Give me 1 minute to give my instructions for people. All right, so we're almost coming to the end. There's still one small little segment that Prof Tan will do later on. But before that, okay, while we finish our symposium today, this is not the end of our celebration. We still have a gala dinner for invited guests, right? So one logistic or housekeeping reminders, those that are going to take the buses, right, to the gala dinner later, please assemble at MD11, the roundabout, at 5:50. Okay, please. 5:50. 50, yes. Oh. Yes. So, all right. So please be there. We will leave without you. Okay, but you always have Grab. Okay, so the final segment, let me invite Prof Tan to give the closing remark.
Thank you, Chin-Wen. Yeah, what a wonderful day. I feel so happy. When things work out, you feel there's a flow. But I think there's a lot of flow today. Everything's felt very smooth and wonderful speakers, great logistics, great planning. And you heard the saying that it takes a village to raise a child. It's also true that it takes a village to organize a symposium, right? Everyone has a role to play. But in particular, I'd like to thank the people behind the scenes, right? Please join me in thanking the following people. Our MC, Chin-Wen, Madhu and Dylan, right in the corner there. Please join me in thanking them for keeping things in order. And at the back, we have my logistics team led by Chente and Fatima. They did so much to get everything up and running. So please thank Fatima and Chente. Thank you so much. Last but not least, let me thank our speakers again, our local speakers, our alumni, and our overseas speakers. Thank you so much for making our event special. Thank you once again. Yeah. So I'd just like to close today's symposium by thanking all of you and to say that for many of us, the night is still young, right? We're going to have a great party tonight. So I'll see you at Shangri-La. I'm going there for a cocktail ahead of time, so you can join me if you want. Thank you.